Good morning and welcome to today's public workshop, Pharmacodynamic Biomarkers for Biosimilar Development and Approval. I'm Mark McClellan. I'm the director of the Duke Margolis Center for Health Policy and pleased to be with you hosting this two-day workshop in cooperation with the FDA. We're glad you're with us. We have the privilege of working on a lot of important cutting-edge regulatory science topics with the agency, and this is definitely one of them, a very timely meeting. A decade ago, Congress created the 351K BLA pathway for biosimilars, but today a limited number of biosimilars have been approved, and the opportunity for patients to get improved access to cost-effective biologic medicines has also been limited. Over the next few days, we're going to hear about biosimilars and the opportunity to encourage additional, perhaps more efficient biosimilar development and approval through efforts to advance pharmacodynamic biomarkers, also called PD biomarkers. We're going to hear from FDA, from sponsors, from academic scientists, from regulators, including from Europe, Canada, and Japan. This broad set of stakeholders will discuss how PD biomarkers might streamline biodevelopment and approval, as well as efforts to identify and advance PD biomarkers and increasing real-world evidence with PD biomarkers. And finally, what healthcare providers and regulators might expect of biosimilars approved based on PD biomarkers, how that pathway might evolve. And go to the next slide. So we're starting out with the welcome and opening remarks right now. Just looking ahead to the meeting agenda in session one today, we're going to hear broad perspectives from the FDA and industry regarding biosimilar development and opportunities to better leverage the tools that clinical pharmacology has to offer to impact biosimilar development and make it more efficient. In session two, we'll hear more detail about the FDA's ongoing efforts to advance PD biomarkers, as well as reactions and additional perspectives from industry experts. Then tomorrow, we have session three and session four. Session three and four will focus on emerging experience with PD biomarkers. The speakers are going to talk about PD biomarkers real-world evidence to date, including how the biomarkers are assessed, circumstances involving increasingly complex clinical pharmacology, how PD biomarkers contribute additional value to clinical development, and how clinicians view biosimilars and the data supporting their approval. Then finally, in session five, we'll hear from the FDA and international regulators as they compare their regulatory thinking and approaches from a range of these global regulatory agencies and discuss the themes that they've heard throughout the workshop sessions and some of the implications. In just a moment, we're going to hear a pre-recorded set of opening remarks from Acting Commissioner of Food and Drugs, Dr. Janet Woodcock. But first, I wanted to go through our usual disclosures and some comments on our virtual meeting format. Duke Margolis aims to serve as a neutral third-party convener. We welcome, in fact, we require a range of perspectives. Our academic statement of independence means that neither Duke nor the Margolis Center take partisan positions, but of course, individual participants here are free to speak their minds and express their opinions regarding important issues. In the next slide, the usual antitrust cautions. Our antitrust policy is described here on our website. I want to remind all of our speakers and viewers that we will not be discussing any confidential, proprietary, or competitive information as part of today's and tomorrow's meeting. And then finally, as many of you know, I think I'm an independent board member at Johnson & Johnson and at Cigna. So next on the virtual meeting reminders, just some brief comments about the format. We're looking forward to a lively and engaging set of discussions. We want all of you to contribute to the workshop. You can do that through the Zoom Q&A function. That should be at the bottom of the screen for most of you. That can be used to include questions or provide a comment or additional insights for the topics of the meeting. Many of our sessions include presentations, but they also include moderated panel discussions and time to address questions from the audience as well. So don't hesitate to submit those questions and comments using the Q&A function. During our panel discussions, we ask that our panelists unmute their microphones and turn their cameras on. Aside from that, we ask our panelists keep their microphones muted, just to preserve the quality of the audio. And finally, our team at Duke will be advancing slides for all you panelists today. So if you are presenting with slides, just say next and we'll take care of that. And again, all of you, thanks so much for joining us. And with that, I'm very pleased to start us off with opening remarks from Acting Commissioner of Food and Drugs, Dr. Janet Woodcock.
Good morning. Thank you, Mark. I'm delighted to be with you to help kick off this workshop and focus on this very important and timely topic. You've convened an impressive group of scholars and scientists, including many from the FDA. When Congress enacted the Biologics Price Competition and Innovation Act of 2009, it was a potential game changer in patient care and for our continuing efforts to support innovation and at the same time increase access to potentially life-saving treatments while reducing healthcare costs. The addition of the 351(k) biologics license application pathway to the Public Health Service Act, which established an abbreviated approval pathway for the licensure of biosimilar and interchangeable biological products, opened the door for drug developers to better leverage the safety and efficacy data of reference products to support the approval of biosimilars. This, in turn, enhanced the potential for greater access to cost-effective alternatives to approved biologic products, which are used to treat, of course, many serious and life-threatening diseases. Unfortunately, as we know all too well, many of these therapies are very expensive. In some ways, our work on biosimilars offers a sequel to the story we've written about generic drugs. But even as the details of the plot are in some ways similar, many are quite different, as there are unique challenges relating to the establishment of biosimilarity. At the FDA, we're studying a number of promising areas of focus that we hope will overcome these challenges. As the biosimilar development community is aware, biosimilar sponsors are encouraged to use a stepwise approach to demonstrate biosimilarity. This begins with the establishment of an analytically highly similar foundation from comparative analytical studies, followed by considering the role of animal data. Then, sponsors should perform comparative human PK and PD studies if a relevant PD measure is available, and compare the clinical immunogenicity of the two products in an appropriate study population. If residual uncertainty remains about whether there are no clinically meaningful differences between a biosimilar and its reference product, biosimilar sponsors have conducted comparative clinical efficacy studies. These studies are time and resource intensive, and many have been larger and more costly than pivotal trials for new molecular entities. This has been done for most approved biosimilar products. One area we're increasing our studying is how to advance the science around PD biomarkers to increase their use in human PK/PD biosimilarity studies to address residual uncertainty. That can decrease the need for comparative clinical efficacy studies. As you understand, PD biomarkers can demonstrate the characteristics of a biosimilar's target effects. This means they can help reduce residual uncertainty about whether there are no clinically meaningful differences between a biosimilar and its reference product. PK data and PD biomarker data are often more sensitive than clinical efficacy endpoints in detecting differences between biosimilars and their reference products, if differences exist. Consequently, the use of PD biomarkers in biosimilar clinical development programs may help streamline and further abbreviate the clinical programs. They're also easier and less costly to obtain than traditional clinical endpoints. On this point, it's important to note that PD biomarkers for biosimilar development are not required to reflect clinical efficacy or tie to the efficacy endpoints. There are, however, critical characteristics of a PD biomarker that make it best fit for use in PD biosimilarity studies to address residual uncertainties between the biosimilar and reference products. And this is what will be discussed in detail during the workshop. The challenge is that there have been relatively few instances where PD biomarkers have been utilized as a pathway for biosimilar approval without comparative clinical efficacy studies. Indeed, among the 30 approved biosimilars, the only development programs that have utilized PD biomarkers are insulin glargine, pegfilgrastim, filgrastim, and erythropoietin, which account for a total of 8 products approved. The FDA Biosimilars Action Plan offers the potential for the FDA to help advance science and provide regulatory clarity on the use of PD biomarkers to demonstrate biosimilarity. We're conducting targeted research and clinical studies to fill information gaps and identify opportunities to increase the utilization of PD biomarkers, decreasing the need for comparative clinical efficacy studies. We're counting on feedback from pharmaceutical academics, practitioners, and other external stakeholders on these efforts, as well as from other experiences to help us move forward. The field of biosimilar development offers enormous opportunities for innovation in the development of new, more affordable treatments. These opportunities are accompanied by great challenges. The FDA is committed to supporting innovation and to making this market even more robust and competitive. In this way, we can support the continued development of safe, effective, and accessible biosimilar products that benefit patients and public health. I certainly hope you have a productive meeting.
Great. Many thanks to Dr. Woodcock for those opening remarks that highlighted the very important issues we're going to be discussing today and tomorrow. And now we're going to get right into that with our first session. This is a large session that's going to last till noon, but we're going to get a large break after that. And this session is on biosimilar development paradigms, current and future perspectives. I'm pleased to welcome our presenters. They include Sarah Yum, Director of the Office of Therapeutic Biologics and Biosimilars in the Office of New Drugs at the FDA, who's going to present an overview of the FDA's perspective and activities. Then Yaoming Wang, who is Associate Director for Biosimilars and Therapeutic Biologics, who will present on clinical pharmacology principles for PD biomarker and biosimilar development. She'll be followed by Leah Christie, Executive Director of Global Biosimilars Regulatory Affairs and Regulatory and R&D Policy at Amgen, and then by Abhijit Burway, who's Chief Medical Officer at Viatris. Following their presentations, I'm going to facilitate a discussion. We're also going to be joined by some additional speakers, including Elena Wolfholtz, who's Chair of Biosimilar Medical Products Working Party of CHMP at the European Medicines Agency, and Isam Zine, who's Director of the Office of Clinical Pharmacology at FDA. So a great range of speakers. I want to thank them all for taking time to be with us today. And we're going to go first to Sarah for her opening presentation.
Thanks, Dr. McClellan. Thank you for the opportunity to speak with you all today about current and future perspectives on biosimilar development. Next, please. I just wanted to start off with a bit of background as context. So over the past 37 years since Hatch-Waxman, prescribers and patients have become quite familiar with the abbreviated pathway for small molecule drugs, and the majority of them are comfortable prescribing and receiving generics. And I've outlined on this slide sort of a comparison of the generic and biosimilar pathway assessments. And by contrast, the abbreviated pathway for biologics starts off from the beginning more complicated by having 2 categories of assessment standards, i.e. one for biosimilarity and one for interchangeability, and only one of which, interchangeable, may be substituted without the intervention of the healthcare provider. And certainly when you read the statute, there is more uncertainty and more flexibility in the statutory language and potential expectations per product. Next slide, please. Now, another major difference is, with generics, you have your pharmaceutical equivalence assessment, and then you have your basically exposure assessment or clinical pharmacology assessment. On the other hand, an abbreviated 351K BLA will look a lot like a standalone 351A BLA with just a difference in proportions, but having the same elements. Next slide. Is that warranted in terms of the additional complexity or expectations? I want to take you through the details a little bit because I think educating our prescribers and patients is going to be really essential to the overall success of this pathway. So this slide is an example of a typical small molecule drug where you think about a dose being maybe in this case a pill, may have different looking containers between the brand and the generic, but both those containers will contain identical molecules of active ingredient. And so here we're drawing the analogy between a molecule and a jelly bean, and then just bags full of yellow jelly beans for either the brand or generic. Next slide, please. By contrast, biologics have way more molecular variety. They start off with the same underlying protein, but then of course, you get the additional changes that occur to the protein in the cell. A big example of that is glycosylation, which does occur in a very patterned way, but does result in millions of slightly different variants of the protein in a sampling of the biologic. So using the same analogy of jelly beans, what we end up with is many different colored jelly beans in addition to trying to copy just one jelly bean. And the numbers of each color of jelly bean are in a dose are never going to be exactly the same, whether you're talking about a reference product or a biosimilar. Now, controlling the mix and keeping it consistent is a fundamental aspect of biologic manufacturing. But we have a lot of accrued knowledge on how to do that, and also the clinical implications of various variants. Because monoclonal antibodies have been very successfully manufactured and marketed since the 90s. So it's not as mysterious and unpredictable as some may think it is. Next slide, please. As complex as this pathway has the potential to be, we are getting the hang of it. We just approved the first ranibizumab biosimilar on Friday. And this means we're up to 31 approved biosimilars, including the first interchangeable biosimilar, which is an interchangeable insulin glargine. 20 of these 31 approved or marketed. Next slide, please. So where are we in our stages of development? I'm thinking maybe we are in the teenage years. We still have a little growing and learning to do before we get to sort of the fully mature or even honeymoon stage of adulthood. Next slide, please. So we still have a lot of work to do, and our goal is to facilitate high quality, safe and effective biologic therapies being accessible for more patients. And our action plans still follow along the previously identified key areas in the Biosimilar Action Plan that was published in July of 2018. And these basic areas include improving the efficiency of the product development and approval process, maximizing scientific and regulatory clarity for the product development community, education and outreach to improve understanding among patients, clinicians, and payers, and then supporting market competition by reducing gaming of FDA requirements or other anti-competitive behavior. Next slide, please. Today's workshop focuses on the very important potential of using pharmacodynamic markers, which are tailored to biosimilar development. And this is going to be a really important mile marker on our journey to a more efficient product development and approval process, and was called out in the Biosimilar Action Plan. Next slide, please. But we continue with efforts supporting all the key elements, whether it be guidances, updating the Purple Book to list patents, all the things that you see here. Next slide, please. Developing and creating more educational materials, media, and a healthcare provider curriculum, which we're working very hard on. Next slide, please. Or continuing to have our regular meetings with the FTC and supporting that collaboration to try and support market competition. Next slide, please. But development and approval is only part of the story, and we obviously need to be able to bring the users along with the story. And we need to be able to tell them the story in a way that makes sense to different people and encourages others to tell the story as well. Next slide. So where we want to go, I think, is clear, but the road to getting there has a number of challenges. One major challenge is the US healthcare ecosystem and economic structure, but obviously this is something outside FDA's purview, and I'm not going to even try to talk about the challenges there. Another major challenge is the fact that the predominant drug development paradigm in everyone's mind is the new drug development paradigm. It predominates thinking, it's more intuitive. You have this direct relationship between data and confidence. That is, the more data you have, the more confidence you have in a conclusion. And then finally, educating people is not straightforward. Behavioral psychology would suggest, and the pandemic has shown, that just because something makes sense doesn't mean people will believe it, even if you have data. Particularly if there are factors triggering a system one type response, i.e. the quick, intuitive, instinctive reactions as mentioned by Daniel Kahneman in his book, Thinking Fast and Slow. Next slide, please. And in that context, we're sort of plopping the biosimilar development paradigm, which flips the data arrow downward by design, and yet we want to keep the confidence arrow pointing upward. How do we make people comfortable with less of the data that they're familiar with, the clinical data, yet keeping their confidence level high? We've got to make them feel more comfortable, and that's a difficult thing. Next slide. What can we tell people that will make them feel more confident with a development paradigm that relies the most on data they know little about and find hard to understand? Obviously, to a lot of patients and prescribers and external folks, basically the development pathways kind of look like this, which is, reference product had all these clinical studies, and the biosimilar has much fewer clinical studies. Next slide. If you think education alone will fix it, I think we'll be disappointed. There's surprisingly little data, but a Cochrane review that was done in 2019 identified one example of a single controlled before and after study in the US that I think is very informative. This study looked at the effectiveness of an educational program versus interventions to regulate drug reimbursement in order to impact prescribing practices. And this was actually a well-done study of 4 different but similarly situated HMOs. So starting on the right with HMOD, which did no interventions at all, it sort of served as a control. You can see there were small changes in prescribing toward a preferred drug and away from the less preferred drug, along the lines of 2 to 4% there. With HMOC, prescribers were provided educational information and coupons, which could be used to reduce patient costs. However, changes in prescribing practices were still small, slightly better than with no intervention, but not by much. HMOB used the approach of educating both prescribers and HMO members, and additionally sent coupons to HMO members directly. And this resulted in a much larger shift in prescribing practices toward the preferred drug and away from the less preferred drug, suggesting that enlisting patient stakeholders, the people who are going to receive the drugs, is an effective approach. HMOA instituted coverage limitations for the non-preferred product, along with full reimbursement and coupons to encourage prescribing of the preferred product. And these interventions were by far the most effective at changing prescribing practices. There was almost a 50% change in the desired directions. Next slide. We can see also that with our early experience in biosimilars, that more than just giving people education is needed. For successful organizations like Kaiser Permanente, which actually had a very high rate of biosimilar uptake and use, educational materials and guidelines were part of the approach, but there was much more. There was involvement of the prescribers and pharmacists in the decision-making process, which included review available evidence and post-market data, also included peer-to-peer sharing of experience, and then also individualized attention, pharmacists, drug education coordinators who could counteract misinformation and answer any questions that patients or prescribers had. Next slide. Ultimately, to get where we want to go and achieve public health goals, we must balance efficient development with increasing confidence in and uptake of biosimilars. Typically, healthcare providers are most familiar with and reassured by clinical efficacy and safety data. And education about key scientific and regulatory concepts regarding biosimilars is not easy to convey or ingest. Efficient development won't help if it makes prescribers less confident in or less likely to use the products. So approach and timing are really important. And finally, education is necessary, but it's not going to be sufficient to change feelings, beliefs, and behaviors, which are a complex issue, but going to be critical in achieving public health goals. If we want this pathway to succeed and increase access for patients, then we'll all need to work together to make that happen. And I think the final slide is basically just links to resources. I thank you for your attention this morning.
Great, Sarah, thanks for your presentation. Great overview, and thanks for the resources as well. We'll try to make sure links to those are available. Next up is Yao Ming. And Yao Ming, I think we can hear you.
Okay, great. I'm unable to turn on the video at this point, but I will begin.
Okay.
In this presentation, I will discuss the clinical pharmacology principles for selecting PD biomarkers for biosimilar development. Good. My video comes back. This is my disclaimer. My presentation represents my personal opinions. So in this presentation, I will begin with a brief introduction of biomarkers and the context of use to help us zoom into PD biomarkers. Next, I'll describe the role of PD biomarkers in biologics and biosimilar development. Then I'll walk through the 5 characteristics of PD biomarkers in FDA's guidance using some practical review examples. After that, I'll try to make a case for the opportunities to increase utilization of PD biomarkers, and finally, I'll end with a summary of key messages. So listed here is the best resource document, the type of biomarkers covered, and each of them is defined by the context of use. Today, we are focusing on the PD response biomarkers, which is the best document defines it as a biomarker used to show that a biological response has occurred after exposure to a medical product. As you can see, at the bottom, validated surrogate endpoint is listed as a separate item, which could also be considered for PD biomarkers as well. We actually have a lot of experience with PD biomarkers in drug development. We have used them to characterize the pharmacological activity and to identify the pharmacologically active dose, the PAD, or the MABEL, as we know them. We use them for quantitative evaluations of dose response and exposure response relationship. Further, PD biomarker data are almost always collected in the efficacy and safety trials as secondary or exploratory endpoints. In certain cases, the PD biomarkers may be surrogate endpoints for assessing clinical efficacy. The role of clinical pharmacology data differs between the standalone BLA and biosimilar BLA because these programs have different goals. For standalone BLAs, we characterize the PK and PD to evaluate the relationship between dose and exposure, dose and response, or exposure and response. These evaluations contribute to the dose selection for clinical trials and for labeling as well. For biosimilar programs, on the other hand, we compare PK and PD between products. The goal is to demonstrate similar PK and similar PD if there are available PD biomarkers. With the foundation of analytical similarity, similar PK and PD can support biosimilarity without comparative clinical efficacy data. This flowchart illustrates 2 approaches to support biosimilar approval when systemic PK is available. The specific approach to take depends on whether there are suitable PD biomarkers or not. When there are no suitable PD biomarkers, the approach 1 will be taken, and this approach includes PK similarity data plus comparative efficacy and safety data. When there are suitable biomarkers, approach 2 will be taken, which includes PK and PD similarity data plus the comparative safety data. Here, the safety data are referring to the immunogenicity data. When you compare these 2 approaches, it is apparent that the PD similarity data are used in lieu of comparative efficacy data to support biosimilar approvals. As of now, we have 31 biosimilars approved. 30 of them have systemic exposure. So among these 30 biosimilars, they are in reference to 10 reference products, and the majority, which is 22 out of 30, have taken approach 1 that included comparative efficacy data. The bar chart here represents a breakdown of the approved biosimilars by the reference product. The pink bars are those 22 programs that conducted comparative clinical study, or CCS, and the blue bars are those with PK and PD studies. It clearly, so far our experience actually indicates that biosimilar programs with PK and PD studies are more efficient. For example, the sample size is much larger for the CCS compared to PK and PD similarity study, and the study duration is much longer for CCS. This table shows the average sample size for the CCS compared to the PK/PD study by their reference product. It clearly shows that CCS have a much larger sample size. This slide shows a second comparison, which is only possible because some of those biosimilar programs that took PK and PD approach also contained a comparative clinical study, although they were not required for the BLA approval. So again, you can see that the comparison also shows the CCS has a much larger sample size. Next, I will move on to the 5 essential characteristics to consider in selecting biomarkers that were described in the FDA's guidance. The first one relates to the temporal change after dosing from the onset of the response to the return to the baseline. And the second one relates to the dynamic range of PD response over the concentration range. The third one is the PD biomarker sensitivity to the differences between products. And the fourth one is the relevance to the drug's mechanism of action. Last but not least is the analytical validity of the assay. For this item, the FDA has a method validation guidance, and there is a very well-established understanding that the reliability of PK and PD data depends on the quality of the assay used. So I will not further elaborate. We will walk through the first 4 in order now. So the first characteristic of PD biomarker to consider is the temporal change, which is showing the time of onset and its return to baseline. The graphs are examples of temporal peak profiles of PD biomarkers for approved biosimilars. They all show a clear onset, except for one. All of them show a clear rise and fall. These PD data were from a single dose study or a multiple dose study. The PD comparisons were based on the area under the effect curve, AUEC, and the maximum effect. Both of them are measured as the change from the baseline. The second characteristic is about the dynamic range, which should be wide enough over the range of observed drug concentration. The figure here illustrates the concept. You can see the glucose infusion rate increases with increasing dose of insulin glargine, indicating a wide range of dynamic response. The third characteristic is the sensitivity of PD biomarker to detect differences between products. This can be achieved by identifying a dose that's at the sensitive part of the dose response curve, which is the steep part. In other words, we want to choose a dose that's not at the plateau region. An example of a dose response curve from the literature is shown here. To evaluate these 2 characteristics, data from a range of doses are generally needed. So a correlation between PD response and the clinical outcome is beneficial, but not required. When feasible, a correlation analysis could be supportive. And here's an example. As we know, filgrastim treatment reduces the time to neutrophil recovery and the duration of severe neutropenia in patients who undergo chemotherapy. The dark line in the figure shows a correlation between the PD response on the x-axis and the clinical outcome measure on the y-axis. Here, the PD response is the difference in AUEC of the absolute neutrophil count, and the clinical outcome measure is the difference in duration of severe neutropenia. The small rectangle in the center marks a plus minus 20% difference in PD response, which correlates to a difference of 0.2 days in duration of severe neutropenia. The finding further supports the sensitivity of absolute neutrophil count for detecting clinically meaningful differences. The fourth characteristic is the relevance of PD biomarkers to the drug's mechanism of action. A biomarker that is relevant to the mechanism of action will measure the PD responses that are effectively demonstrating the characteristics of drug's target effect. Along this line, the guidance further elaborates and explains that a panel of biomarkers can be considered. These characteristics may be easy to address for some products, such as the approved biosimilars for filgrastim, pegfilgrastim, erythropoietin alpha, and insulin. The blood cells and glucose in these panels were used as PD biomarkers. They clearly reflect the mechanism of action as shown in the figure, and they were used for evaluating efficacy in the innovators program. On the other hand, it may be more challenging to for other products with more complex pharmacological effects and less well-characterized mechanisms of action. In such cases, it may take a bit more work to sort out available PD biomarkers and evaluate whether or not they're suitable for biosimilar development. So how do we identify the relevant PD biomarkers for biosimilar development? One can argue that published literature contains abundant helpful information. When there is a good understanding of the mechanism of action for the reference product, it may reveal opportunities for multiple PD biomarkers. Conceptually, in this figure, it shows that when a drug binds to a receptor, the binding event triggers a cascade of biological response. They can be measured as changes in the PD biomarker level. And these changes could be considered as reflecting the drug's mechanism of action. Some of them may be related to the efficacy. Some of them may relate to other effects. So far, approved biosimilars have used biomarkers that are tied to the clinical efficacy. But this type of biomarkers are really just the low-hanging fruits. For PD biomarkers in drug to be used in biosimilar development, they're not required to reflect clinical efficacy. Therefore, it represents an opportunity to explore PD biomarkers that previously showed a dose-response relationship. After identifying PD biomarkers that could be relevant to the mechanism of action, a pilot study may be necessary to further fill in the knowledge gaps and assess the suitability with respect to the other characteristics. A pilot study that evaluates temporal PK and PD profiles across a range of doses would be most helpful. It could generate data that illustrates the PK and PD variability and the dose response relationship. These data can also enable further use of PK/PD modeling to evaluate the sensitivity of PD biomarkers and optimize the study design. With the pilot study, one can gain experience with the assay for the PD biomarker and further refine the assay for later use. So in summary, the underlying clinical pharmacology principle is that a similar PK and PD will provide similar efficacy and safety. Therefore, using PK/PD similarity data can remove the need for a comparative efficacy study. And that could lead to a more efficient biosimilar program. The FDA's guidance recommends 5 characteristics for selecting PD biomarkers. Again, they could be a single or multiple PD biomarkers for similarity assessment. Very importantly, the PD biomarkers for biosimilar development are not required to reflect clinical efficacy or tied to the clinical endpoint. There's an opportunity to explore PD biomarkers that show dose-response relationship. Pilot studies, as well as modeling and simulation, can be considered to provide justifications for PD biomarker selection and facilitate study design. Lastly, I would like to acknowledge all my FDA colleagues who have contributed to the review of biosimilar submission and the development of scientific policies. Thank you. With that, I close my presentation.
Right, Yamin, thank you very much. Again, covering a lot of ground. We really appreciate it. And next, I'd like to turn to a couple of industry expert perspectives, starting with Leah.
Thank you very much. Thank you for the kind invitation to come and speak with this group today through the conference. I'm looking forward to a lot of good discussion here. So again, my name is Leah Crissell. I am the head of Global Biosimilars Regulatory Affairs and Regulatory R&D Policy at Amgen. And I'm going to spend a little bit of time giving one industry perspective around the role of pharmacodynamic biomarkers for biosimilar development and approval, really more from the regulatory policy perspective and what it is that we're thinking about, where there are maybe some gaps that need to be addressed, some of the considerations, and how we can best move forward. Next slide. So we've already heard from the previous presentation from Dr. Yim about how stakeholders view the importance of the regulatory approval standards for biosimilars and really how it is that they're looking at the data that's involved with supporting that approval. So we know that scientifically sound and robust standards for approval and manufacturing for biosimilars are really critical to stakeholders and help to support acceptance of biosimilar and interchangeable products, uptake and use of these products. So it is important that as we think about this and we talk about these scientific aspects, from a regulatory perspective, it is important that we're making sure that we're conveying that even as we're expanding and pushing the envelope on the scientific side, that we're really maintaining appropriate regulatory approval standards, and that these are scientifically sound, they are robust. We don't want any sort of backsliding based on the ground that we've been able to gain in terms of biosimilar uptake and approval. Next slide. So we all know that the analytical similarity assessment to support that the biosimilar is highly similar to the reference product is what we refer to as the foundation of a biosimilar development program. So there's a lot of advances that we're seeing in this scientific space, and we know that new analytical technologies that are emerging, continue to emerge over time, can provide additional informative data about structural and functional similarity and really build that foundation of a biosimilar development program. We're also seeing that advances in the scientific area specific to in vitro and in silico technologies can potentially help to target the clinical testing that's needed to support biosimilarity, resolve uncertainties that's seen in the analytical testing, again, really help to understand not just the structural and functional aspects when looking at similarity, but what role structure and function plays in terms of clinical performance. But we don't have all those answers. Analytics can't tell us everything. And there's also a spectrum of products that we're talking about in the biological space, from products that are well characterized, well understood, also fairly simple biological molecules, all the way up to ones that are more complex. And we are seeing more complex biologics coming on the market in terms of what will ultimately be reference products. And so we're going to have additional complexities over time for biosimilars, moving into things like antibody drug conjugates, bispecific antibodies, other types of more complex biologics. So it's important to consider what the analytical exercise can tell you and what uncertainties you have moving out of that. And so, right now, comparative clinical testing is still needed to demonstrate that there's no clinically meaningful differences for a number of the products, maybe not all the products, but for a number of products. And so with that, we want to think about, well, how is it that can we improve efficiencies in a biosimilar development program, recognizing coming out of this analytical exercise, we still will have residual uncertainties for many of the products. Next slide. So as has been discussed already, we know in FDA guidance that they stated that in certain circumstances, biosimilars can be approved based on PK and PD biomarker data without a comparative clinical study using what we refer to as more traditional efficacy endpoints. Of course, we also need to keep in mind the need for collecting appropriate safety data, including immunogenicity data where that's relevant. So from this guidance, we have the quote here that, again, is really focusing on the fact that we can use clinical pharmacokinetic and pharmacodynamic data to demonstrate that there's similar exposure and response between that proposed biosimilar product and the reference product. And that data can be potentially sufficient to completely assess whether there's clinically meaningful differences between the products, again, notwithstanding the need for an adequate assessment of immunogenicity where that's warranted. So we already have a good regulatory framework to be working from. That door is already open, so to speak. But what does this really mean in terms of a development program, and what does it mean from industry side about how it is you move in this space? Next slide. So again, we know that biomarkers can improve development efficiency and sensitivity, but we get right down to it, what is it that we're talking about? There's a big push to improve efficiency in biosimilar development programs. We heard those comments from Dr. Woodcock, and I think that that's a theme carried through this workshop. It's why we're all here to talk about this. So there's a big push again for comparative clinical studies incorporating flexible study design. Where can we push that envelope? But in the aspect of flexible study designs, in this context of talking about PD endpoints, we want to think about population aspects in those studies, such as the use of healthy subjects, but then also the use of these pharmacodynamic endpoints, as well as the PD arrays that Ming has spoken about. And these should be considered when appropriate, when scientifically appropriate. We have the regulatory framework to do this. We have the flexibility. FDA has already, again, opened that door through guidance. So we want to be able to have these conversations, and we should be trying to push the envelope on this. So FDA's goals, as outlined in the Biosimilars Action Plan around this topic, include the development and validation of pharmacodynamic biomarkers that are tailored to biosimilar development, and aspects of in silico modeling and simulation to help evaluate PK and PD response versus clinical response. Those relationships can be looked at using existing clinical data. So the work that FDA has done and continues to do that is outlined in the Biosimilars Action Plan is really critical for this conversation to set the foundation about what's expected in the development program. How can industry move through these conversations in an efficient manner that's really supporting drug development? And we'll talk a little bit more about why that right now is a little bit of a space that we're working through, and sometimes it's not always the best approach. So we know that these approaches where we can have this flexible study design can reduce the size of a study and increase the sensitivity of that comparative clinical study. And I will say, when I talk about comparative clinical studies, I'm talking about whether that's pharmacokinetic, pharmacodynamic, or a more traditional comparative efficacy study. So there's a lot that can happen within this concept of a comparative clinical study. And again, we need to have these conversations about where we can have flexibility to improve the efficiency of biosimilar development programs. Next slide. So we've already heard a little bit about scientific considerations when we talk about pharmacodynamic markers that could adequately support a demonstration of biosimilarity, really, again, focusing on that there's no clinically meaningful differences between the products. So we've talked about this concept of a suitable pharmacodynamic endpoint. Well, what does that really mean? The pharmacodynamic biomarkers that reflect the mechanism of action of the biological product will have greater potential to be more sensitive endpoints for detecting clinically meaningful differences. But we don't always know everything about the mechanism of action. For example, there's been a previous public discussion of a proposed biosimilar that is used across multiple therapeutic areas where the mechanisms of action were described by FDA as known, likely, and plausible. So again, this is why analytics can't tell us everything. When we do these functional studies, we don't always know everything about the critical quality attributes. We don't always know everything about the mechanism of action. And where you don't have, the mechanism of action might be unknown or it's not well understood, you don't have all that information, a PD biomarker is less likely to be reasonably predictive of clinical outcome. So you have this balance there about how much you know, how much you understand, and then looking at what you consider to be, again, this concept of a suitable biomarker. We know that they don't need to be validated surrogates. As was discussed in the previous presentation, you don't always necessarily have to have a clinical correlation in terms of clinical response, but you do have to have an understanding that what it is that you're evaluating is relevant and giving you data that's meaningful in terms of an assessment of whether there's clinically meaningful differences between the products. Another aspect is the sensitivity of the PD biomarker to differences between the proposed biosimilar and the reference product. For any study that you're conducting in the space of a biosimilar, you want to understand that you have sensitivity to detect differences should they exist. You don't want it to be that you have a study where you didn't see any differences between the biosimilar and the reference product and have uncertainty about whether there's truly differences between the products and that's why you didn't see a difference, or if you just didn't have a sensitive PD biomarker. Also, aspects of the dynamic range of the biomarker over the exposure range of the biological product. There's also various methodologic considerations such as assay validity, and all this needs to be built in to the decision to use a PD biomarker or an array. Again, you want to make sure that that study you're conducting will answer the question that you have of whether there's no clinically meaningful differences between the products. Next slide. So from an industry perspective, when you're thinking about biosimilar development and moving through setting out a development program and thinking about what studies you need to do, identifying a novel biomarker or PD array to support a demonstration of biosimilarity can actually turn out to be more resource intensive than conducting a comparative clinical study with traditional efficacy endpoints. So those factors that I outlined previously, about assay validity, looking at exposure response relationships, looking at sensitivity, there's a lot of work that would have to go into that to justify the use of a novel biomarker or a PD array to be able to answer that question about whether there's clinically meaningful differences between the products. It's somewhat difficult to do that within a development program and really stay on time, make sure that the data that you collect is going to support a demonstration of biosimilarity. So where we have a lack of regulatory certainty or predictability for the acceptance of a PD biomarker or an array, it becomes risky compared to conducting a comparative clinical study with a more traditional efficacy endpoint. So there's a lot that needs to go into this from the FDA side as well as industry side when thinking about how to move through this and support a biosimilar development program to make sure that it's timely, it's efficient, but at the end of the day, you're going to have the data that you need to support approval. Next slide. So there's a lot of potential for pharmacodynamic biomarkers and biosimilar development programs. Again, we're seeing new and emerging technologies that are going to enhance biomarker identification and form the analytical strategies and really, again, target our clinical studies and improve the efficiency of development programs. But there's additional scientific exploration that needs to occur outside and in advance of initiating a biosimilar development program in order to really see this come to fruition about improving the efficiency of a biosimilar development program. So there's work that we already know that FDA is doing and will continue to do, and I think we'll have more conversation over the 2 days of this workshop around what constitutes an appropriate PD biomarker, how to identify the biomarkers for biosimilar development, what constitutes a suitable PD biomarker. And it's really important for industry to have regulatory transparency, clarity, and predictability to support these development programs. It's always a race to the finish when you're doing drug development. So having that transparency, clarity, and predictability is really key when we're developing these products. And again, tracking back to what I first said, the success of biosimilars really depends on the continued development and maintenance of scientifically sound and robust standards for approval. So we want to make sure that we're doing what's scientifically appropriate, not conducting studies that really don't need to be conducted, at the same time balancing that we need to make sure that we have appropriate regulatory standards for these products in order to ensure that what we're approving are safe and effective biosimilars for patients, and we can also maintain the confidence in what it is that we're doing for these products. Thank you.
Great. Thanks very much, Aaliyah, for that presentation. And next, I'd like to turn to Abhijit.
Thank you. My name is Abhijit Barve. I'm the Chief Medical Officer for Viatris. What I'm going to be talking about over the next 10 minutes or so is really the learnings from biosimilar development from an industry perspective over the last decade and the role of PD biomarkers in product approval. Next slide, please. Again, this is the disclaimer. These views presented are my views and do not necessarily reflect that of our organization. Next slide, please. I know this pyramid has been shown multiple times, and I always joke with my colleagues saying that this is probably the most widely used pyramid in drug development. But I think there's some key factors here that we need to talk about when we talk about this stepwise approach. And from our perspective, I think sometimes we get lost into it in terms of the overarching philosophy at each step of the process really is to eliminate residual uncertainty. And over the past 10 years or so, or maybe, since we have learned a lot, especially as it, relates to that top part of the pyramid, which is really the focus of our discussion here in terms of how we can make it efficient. As sponsors, we have a very good understanding of how that comparative efficacy study design looks like today. We know how to do meta-analysis of multiple studies, especially for products, where a lot of data was available. And we'll talk about some of the limitations going forward in terms of, in the past, we were able to do that. We were able to do a robust meta-analysis, 5, 6, 7 studies, do all kinds of statistical calculations to calculate that equivalence margin. We know how to, satisfy the requirements of different regulators, ratios in case of FDA differences for EMA, and then come up with a sample size. And there are lots of, successes with this particular approach. And the goal here is to what we have, what are the challenges we will see going forward. And has been the focus of this workshop. It's really, in some cases, the use of these comparative pharmacological studies and clinical assessment of immunogenicity may provide. So you don't have to do the comparative efficacy studies. Having said that, I think this conventional biosimilar paradigm is still relevant. We'll still have, because only in some cases we can truly meet that requirement of the 5-point criteria that we will talk about throughout the next 2 days in terms of what is there. So it is still relevant. The conventional biosimilar paradigm is still relevant, but I think we need to really apply the learnings over the last 10 years and see how we can take it to that next level, as well as use the scientific advances, as some of the other speakers have said. Next slide, please. I'll spend a minute or two on the evolution of the insulin regulatory pathway. And the reason for this is that there's been a lot of discussion in terms of focus in terms of the first interchangeable biosimilar being approved, which happens to be insulin glargine that was approved about a month ago. But if you really look at these products, right, I think the evolution has been really meaningful, as well as it has focused in some of the ways how things have evolved. So prior to 2020, as all of you probably know, that many of these hormones, insulins, as well as some of these smaller proteins were approved under the 505 pathway. So if one had to develop a biosimilar in quotes, or a kind of a similar product for these insulins, one had to follow the 505(b)(2) pathway, which included doing a PK/PD study, a type 1 study, a study in type 1 diabetes patients, type 2 diabetes patients, also involving close to 1,000 patients. So on 23rd of March, these products moved to biologics. So all the products that were approved under this 505(b)(2) moved to 351A, and then to 351K. And then we had the new guidance on insulin interchangeability, which allowed us, or allowed the approval of the first interchangeable guardian. So I think there is a huge regulatory progress in that particular regard. So today, if one has to develop a product which is an insulin or an insulin analog, we have a much abbreviated pathway in terms of what can be achieved. So from that regulatory perspective, I think there is a lot of progress. Next slide, please. So as I said, there has been a significant evolution in the regulatory knowledge as well as expectation. So as FDA and EMA has gained more and more experience assessing biosimilars, the approach has been much more flexible. As was mentioned today, I think, we know there are some unique challenges. We can't always measure PK in some of these studies. So for ophthalmic biologic, the first one that was approved recently, or biosimilar, we may not be able to assess systemic PK the way we do it for other products. And the regulators have been flexible with this regard. Also, with regards to, in 2018, we had the PEG biosimilar being approved without conduct of confirmatory efficacy study, just based on the PK and the biomarkers. So there has been a lot of progress in that particular arena, as well as was being shown with the recent approval of the interchangeable insulin products. But I think the challenge that the industry is facing really is that we don't have suitable biomarkers that would always satisfy all the 5-point criteria. Whether it be monoclonal antibodies, whether it be fusion proteins, with the recent ones, as well as we will have to assess immunogenicity, which will still require some degree of safety efficacy in patients for most biologics. So as I indicated, the conventional paradigm is still applicable. I think we need to find ways how we can adapt that particular paradigm and see how we can accelerate the development of these specific products where there may not be suitable biomarkers. And I'm sure the next 2 days there will be lots of interesting discussions around that topic. Next slide, please. So I think it comes to a question that we keep asking ourselves, do the classical comparative efficacy study, what kind of value do they add? And this was a publication recently that came out where there was a systemic review of 38 studies that were done. And each of those studies, and again, this was what was available in public domain, and this followed a robust CMC as well as human PK data that demonstrated comparability. And in each of those 38 studies, the comparable efficacy was always confirmed. So, we designed, these studies were designed as per what I mentioned, the meta-analysis, equivalence margins, etc. And each of those 38 biosimilar programs, the comparative efficacy was confirmed. The 2 programs where it did not was really more focused on higher immunogenicity that was detected. And these were relatively earlier programs. When I say them in the last decade, there are some programs that are earlier where we were still learning about CQAs and their impact on certain aspects. So once these things were corrected as part of the process, I think even these 2 products followed that path. But this is what is available in public domain. I think one of the things that should happen is really for the regulators to look at, because they have got access to much more data, because sometimes people always say that failed studies don't get published. So really to take a look at what are we learning from these comparative efficacy studies? Are we really adding significant value, or the ones that are designed appropriately with all the checks and balances in place? And I think that is an important aspect that we also need to look at as we move along this journey for efficient development of biosimilars. Next slide, please. So as I said, when you talk about the learnings, we know that the analytical methods are far, far superior, even when we started a decade ago, right? I mean, we are able to detect things, or things which even sometimes the innovator or the comparable product did not develop it, because that was developed maybe 15 years ago. So we have ability to detect small differences, whether it be in the physicochemical profile, that base of the pyramid we keep talking about, that being solid. We talk about receptor binding with respect to various bioactivities. I think we have got a very strong understanding of the critical quality attributes. All of us have gone through this process of critical quality attributes. How do we rank them? How do we, how many batches are needed to show similarity, et cetera. We have gone through that exercise over the past 6 to 7 years. And I feel, and we have got this very good understanding of what these critical attributes are, whether it be being the glycosylation that Dr. Ian mentioned about the jelly beans, slightly different. We know what those are, how they impact PK, how they impact safety, how we can manage them, how we can control them. We know all of that. We know that clinical PK studies, when we are able to measure them, are highly sensitive to detect product differences. So there is a lot that we have learned in this space as it relates to CQAs in terms of the physicochemical and biological characterization. And we really need to think, what should be that study that is needed from that traditional efficacy study, and what really, in terms of the residual uncertainty, it is addressing. And I think that here the role of looking at the PK and PD biomarkers is going to be extremely critical, along with what we know in terms of CQAs, the CMC, and the analytical capabilities. Next slide, please. So again, this is really the PD biomarkers in action, as I call it, right? We have 4 products that have been approved: insulin glargine, pegfilgrastim, filgrastim, as well as EPO. But this can be applicable to other products, whether it be Darbo, whether it be Aspart, whether it be human insulin, or recombinant human insulin. And here we have got the rule book. We know that each of these PD biomarkers satisfy those 5 key criteria: time of onset, the dynamic range, the sensitivity, mechanism of action that Yao Ming presented here. So we have done that. We have got these products approved. In some cases, immunogenicity is needed. In some cases, immunogenicity is not needed. We have got robust PD markers, whether it be reticulocyte count, whether it be hemoglobin level, whether it be the glucose infusion rate in case of insulins, or looking at AUC. So this is all there. I think that die is cast. We know how to do this. But how do we take this to that next level for products where we may not be able to have these products satisfying those 5 criteria? Next slide, please. So this is going to be the core of the next 2 days, right? Are there alternate pathways possible for products without biomarkers? And before we get into that, I think we have to really look at what the challenges are going to be, over the next couple of years as we continue along this journey of developing biosimilars. First of all, I think we have been comfortable in terms of calculating that sample size for that comparative efficacy study. We had lots of data, let's say for trastuzumab, whether it's bevacizumab, whether it is any of the other fusion protein. There was a lot of data that was there. We could do a nice meta-analysis, which studies to include, which studies not to include. This is going to be incredibly difficult for the newer biologics, what we call as the wave 3 in the industry parlance, in terms of the newer biologics, which are going to go off patent over the next 5 to 7 years. In these cases, if you really look at it, we don't have that luxury of multiple studies. We don't know how that true biologic response is. And if you talk about the space of oncology, this becomes even more difficult, because what we're seeing is that the standard of care is changing. So the meta-analysis of the studies that were done so many years ago, the standard of care might be completely different today. So how do you design studies using that traditional approach of comparative safety and or efficacy studies? Now we have combination of multiple biologics today. The effect size, although small, clinically relevant, and small and clinically relevant, I must mention, is going to have a huge impact on the traditional sample size calculations. So when on one hand, we're talking about efficient biosimilar development, but these are the real challenges we are going to face. We are going to face challenges where many of these products, which have been once a week or once a month, moving to every 3 to 6 months, making immunogenicity assessment even more challenging. So I think that over the next 2 days, I think we really need to think through, do the biomarkers of the prognostic efficacy indicators, do they have to satisfy these 5 criteria outlined in the guidance? And we look at innovative approaches where biomarkers may not be able to satisfy these 5 criteria. We all talk about this orthogonal methods, right? This is kind of the word that has been so often used, where we do different techniques and try to come to that same conclusion. Can we combine orthogonal biological characterization techniques, whether it be dose-dependent binding in vitro, some of the in silico activities, supplemented with some prognostic mechanistic assessment, which we feel are likely to be more sensitive than these large efficacy equivalent studies. So what can be done in this space? And I'm sure there's going to be robust discussion in that regards. And I hope that we come to some, realization, because what I said in that first bullet is real, and folks will start or are already kind of, started to kind of feel in terms of how to look at some of these challenges. Next slide, please. So this slide really summarizes, I think the qualified PD biomarkers have definitely streamlined development. I think today, if we have to develop a product fitting those traditional biomarker category, we know the rule book, and I think this has significantly helped us. But I think we need to take that next step. We need to really know what we have learned from those CQAs. I know we talk about CQAs as if, but there is a lot that has been learned. We know what the impact of certain glycosylation patterns have on PK, what they have on efficacy. We can use all that knowledge as we try to come up with, this orthogonal methods to see what is the best way to move forward. Looking at in vitro PD assays, not necessarily always having that level of the 5-point criteria, because I don't think it may be feasible for certain things. So how can we use some of these exploratory markers that are there, and try to tie them up with what we know with regards to in vitro assessments, and build that bridge towards that efficient biosimilar development. Thank you.
Great. Thank you very much, Abhijit. A great set of presentations. And now we're going to go into some discussion time. Would like to remind you all, hopefully you've got it open, but that you're invited to ask questions or make comments through the Q&A function. And special thanks to our panelists who have been working away at answering those questions. So we appreciate that additional dimension to this webinar. So please keep those coming in. We'll try to get to a number of them during this discussion time that we have. And I'd like to start off the discussion. I mentioned we're going to bring in a couple of other speakers to help with this, and turn first to Isam for your opening remarks. Isam, you lead the Office of Clinical Pharmacology at the FDA. You've seen a lot of the advances in biomarker development impact new drug development, been very important for many areas of clinical development of new drugs. And we just heard, including from Abhijit's presentation just concluded, about some of the challenges and opportunities for streamlining the use of PD biomarkers in biosimilar development. Do you see you make some connections maybe between the FDA program experiences with new drugs and how that could be leveraged for biosimilars?
Sure. Thanks for the question, Mark, and great set of presentations to get us started. We've had several efforts over the years that have had as their main goal to push and to mainstream innovative science and drug development tools into both drug development as well as regulatory evaluation. And when you sort of think about them, we're thinking about programs in quantitative pharmacology, like the end of phase 2A program, which is a bit of a legacy program now, the Fit for Purpose initiative, and of course, the Model-Informed Drug Development Program under PDUFA 6 that I think folks are familiar with. And those are sort of biomarker adjacent programs. When we're thinking more directly in the biomarker space, we've had opportunistic incorporation of biomarkers, genomic biomarkers, for example, in INDs. We've had safe harbor programs, the voluntary exploratory data submissions program, which I think is interesting to think about now in light of some of the challenges that were just raised by the last speaker. And then, of course, biomarker qualification, which morphed into drug development tool qualification and incorporated clinical outcome assessments and animal models. And they all tend to have the same trajectory of this kind of classic hope hype cycle, where some stimulus, whether it be some innovation, some technology, has led to an incredible amount of enthusiasm about the applied science. And then you hit this kind of peak of inflated expectations, and then this plummeting and disillusionment, which I don't think is that bad. I see that as sort of a recalibration of expectations. And it may be worth just thinking about where we are in the PD biomarker space as it relates to biosimilars on that whole kind of trajectory. And there are a few, I guess, convergences or things that all of those programs have in common in terms of this hope hype. In terms of the sort of initial kind of enthusiasm and then a little bit of the recalibration, really, it was the constraints of the science that largely drove that. As we started to apply our understanding of the science, There were a lot of uncertainties around the specific methodologies and approaches that we were using. There was a steep learning curve for non-technical experts, very few instructive cases, and frankly, really high organizational activation energy in terms of trying to integrate these new approaches in already very well established operational frameworks, regulatory frameworks, and this is within the context of both companies and regulatory agencies. And then when you think about this kind of uptick in this new steady state where we did begin to mainstream some of these approaches, it was really due to continued advancement in the science, growing championship among key decision makers and policy makers. And those were really motivated by tangible examples of impact in drug development, in regulatory decision making, and patient care. And one of the key things that can't be understated, which I think is important about today, public engagement and broad discussions were critical. Discussions around use contexts, where the low-hanging fruit are, the high probability of success, what are the evidentiary considerations. And then as we gained more practical experience in all of these programs, we're able to establish workflows internally that allowed for efficiencies in this sort of regulatory drug development interaction. And so when you look at kind of the successful and the sustainable programs, we really had an integrated approach to all of these. They included creating an environment that increases stakeholder acceptance of these approaches. That has a lot to do with increasing familiarity and acceptance in the interdisciplinary team. It's very critical to kind of acknowledge that the nature of kind of how we make decisions in-house, and I'm sure it's the same in companies. Clarity around the issues to be addressed by the biomarker. We found that you have to have sufficient opportunity to vet the approach, including the value proposition of the biomarker, as well as the limitations. And sometimes that happens. We don't have the time in the context of sort of typical milestone meetings to do that. And so to build in that apparatus to have more real-time engagement was really critical. And we did that through a combination of dedicated programs that overlay over existing processes like the MIDD program. The voluntary exploratory data submissions was a standalone incubator, non-regulatory, that allowed companies or consortia to come in and talk to us in a non-binding way to talk about various aspects of biomarker development and use, validation and use. And in some programs were hybrid. So, so that's sort of the kind of cultural and infrastructural operational considerations. One thing that I think is becoming, was clear and is clear now in light of the trends that our last speaker just mentioned, which might limit our ability for a robust sort of single biomarker driven approach, was that we really need scientific, community-driven standards and best practices that lead to this consistent application and evaluation. And these included convergence on not just the science, the best practices in the development and the verification or the validation of the biomarker, but shared methodologies and reporting practices. And to the extent that those can be standardized or templated, that led to a whole lot of increase in efficiency in regulatory interactions. Public workshops like these have to include both subject matter experts and non-technical experts. We actually get a much broader and sort of well-rounded set of considerations and discussions when that happens, and it helps to socialize the concepts among people that are actually going to be making the decisions. And of course, all of that needs to be parlayed into some sort of science policy. In our office, we've been probably for about close to 8 or 9 years now, been working on behind the scenes on product specific, what we call product specific briefs, which are our internal evolving thinking on for a given biosimilar, what might the PK and PD considerations need to be for clinical pharmacology studies or comparative PD studies to be the full stop on development. And then just the final thing I'll leave with is the capacity and expertise. I mean, the education component is big, the knowledge management component is big, And governance has been really important. Many of these successful programs have had standing committees with senior leadership, interdisciplinary in nature, and that has really ensured buy-in from senior leadership, consistency in the decision making, as well as a expression of a wide range of opinions and diversity that allows us to make the most important decisions that the science allows at the time. So I'll stop there, Mark, and give others some airtime, and happy to answer questions as they are.
Great comments. I just do one follow-up question at this point. So you described a process of sort of the evolution of shared knowledge, and not just knowledge, but practical considerations as well. And you did mention that towards the end, we heard this in the comments earlier about in a case of insulin glargine, the development of an FDA guidance document or standards was really helpful. And you talked about eventually this process getting standards as well, and some of the collaborative steps that can make and diversity of perspectives brought together can make that happen faster. Is that kind of where this ends up at the level of some FDA guidance? And is there any other comments you can make about, if so, what's the fastest and most efficient way to get there?
Well, I mean, I guess I'll make a few comments on the guidances in general, without speaking directly to the pipeline, but a few things. One is it may not be recognized by folks outside of the regulatory environment or outside of FDA that we can entertain guidances or guidance-like documents to inform our own thinking from the outside. As you know, Mark, people could submit to the public docket what they think should be the evidentiary considerations or portfolio for either specific biosimilars or specific policy aspects of biosimilar development. So that is not out of the realm of possibility, and we have examples of that where we've incorporated that into new drug development, especially in areas of high unmet medical need. So I think that mechanism needs to be appreciated, and that's a sort of an example of, let's say, crowdsourcing, if you will, the best thinking that might come out of industry, academia, other regulatory, and even individual stakeholders. So that's one piece. In terms of the sort of the internal kind of activities, many, I think that at least within CEDAR, there are at least three that I'm aware of, at least three, maybe more governance committees that focus on biologics, biosimilars related activities, and have done a really nice job kind of connecting the dots among the different offices within the center. And so, several factors go into whether or not we can easily kind of parlay these into guidances, and they have to do largely with precedent and experience. So you have the scientific piece, which hasn't been completely worked out. I mean, we've been talking about orthogonal signatures for close to 10 years, and sort of what does that mean practically? And I think some of the work that you're going to see from our Division of Applied Regulatory Science will speak to how that might be developed. But so you have the scientific development, and then you have to have the knowledge sharing and precedential database, if you will. And so, and then of course, a lot of it is going to be driven by the complexity of the molecule. So not the easiest thing to do, but not not impossible. And I think it's been exciting to see, at least over the last year or so, how that's materialized into some, explicit policy documents.
Great. Thanks for those really helpful perspectives based on a lot of FDA experience. I would like to turn to bring in Elena now. Elena, you've been working extensively on these issues in the EMA context. Wonder if you'd like to comment about what you've heard, any similarities, differences you'd like to highlight in terms of how the regulatory development is taking place in Europe, where there's a longer experience with these biosimilars in general.
Well, yeah, thank you really for having me participate in this conference and also in this very first session. Very much appreciated. Also these excellent presentations that we've heard, bringing us up to speed and really giving a very, very good background. Well, in terms of the European perspective, I would like to just resume that we do have a law and we also have guidelines, as you're aware of, and we have an overarching guideline that specifically speaks to PD markers and their possible role in allowing for the fact that we do not necessarily need a classical comparative clinical efficacy and safety trial, and it lists three particular points, one being that you need a comparable dose response or concentration response relationship. One is that the PD marker should be actually an accepted and validated surrogate marker, which is a surrogate marker for a clinical endpoint. And the third condition being that equivalence margins are mandatory and should be predefined. So with these conditions, then we went along, and as was already discussed here, I think our thinking is very congruent, our experience is congruent, that we have these different product classes where we've really seen and have actually accepted now de facto, but also in our revised guidance documents speak very clear that we do not need efficacy trials, and they've been named before here in this session. So I think that's fine, but there are many other candidates and that we've looked at, and what we've seen really is that perhaps we need to revise our guidance, because we're increasingly struggling with the fact that even if of these 5 points that have been mentioned and that basically we think are very, very important, even if the analytical validity of the PD marker assay is granted and the sensitivity is clear and the dynamic range is clear and so on, yet we may have a problem in that we do not have the means to actually define the equivalence margin because the data is lacking. Or what we also find is that there may not be a one or the one marker where there's actually a very clear correlation already demonstrated with a hard clinical endpoint. So this condition that we still have up in our guidance document as it stands right now, we find is a stumble stone in a way for further acceptance of PD markers. And this is something that again and again we're discussing. In terms of what are we doing, I would like to mention two things, how we now try to evolve with possibly a new guidance. One is something I believe is inevitable, and I really appreciate it that Sarah Yim spoke to the review of available evidence. I mean, what have we learned? We have more than 60 approved biosimilar products, so really a great wealth of products on the market, and we have many scientific advice procedures. I have pre-recorded presentation that you will hear tomorrow on this wealth of experience that we have. But the point is here, we want to write a master product guidance document. So see what have we done so far and really challenge the scientific background that was the basis for the scientific advice that we've given so far. And we may see some adjustments here and there, also taking into account what we've learned now in terms of PD markers for the different products that are on the market, are being developed right now. The second big thing, of course, is the tailored biosimilar pathway. We, as you're aware, have had a pilot procedure here, and we've concluded that this is really a new pathway that we would like to offer to the industry, meaning that we would like to invite applicants to talk to us what are the physical, chemical, and functional assays that you would like to use and engage in terms of a quality characterization, then come back, give us really mature quality package. And if this is looking good, then we would like to discuss a tailored a clinical development plan, meaning that if such development really looks good at the quality stage, possibly we can forego the efficacy, safety, and immunogenicity trial, and rather have the evidence more on the PK and PD clinical study. So here again, I think that the evolution, the reliance on PD markers could and should increase, mostly in addition, of course, to PK work. And so I think these are two very important aspects of our work right now.
Great, Elena, thanks very much. Very helpful discussion. I appreciate all the experience you're bringing to this meeting today and tomorrow. Looking over the Q&As, again, appreciate all of our panelists being so active in answering those. Encourage people to take a look. There were a number following up on the comments made by some of our speakers about where to go next. So clearly some very important progress and now clear guidance in certain areas of biosimilars, but for the more complex, the third wave, the newer ones, most monoclonals, fusion products, question in the chat about some of these as well. There's not so much clarity around suitable biomarkers, and we've touched on already what to do in that circumstance. Abhijit, I think, talked about maybe a modified procedure based on what we know, maybe reducing, if not eliminating, the need for a clinical efficacy study. I wonder if the panelists, if any of the panelists would care to comment further on this topic, since it seems to be one where there's obviously a lot of interest today and a lot of relevance with further biologics development and potential biosimilar availability of these more complex products going forward. Any volunteers? Abhijit, you raised the issues to a good extent.
Yeah, I can talk a little bit, and I think it also ties to what Leah mentioned, right? I mean, in the industry, so biosimilars are not like novel products. So when Nissam talked about developing biomarkers as part of the biomarkers as part of new drug development, it's slightly different or quite different, I would say, because this is usually kind of a race first to market, right? I mean, these are at the end of the day molecules that, so if we don't have a clear path on what is that expectation from the regulators, then the companies are going to take that default step of going with that standard approach of doing that. And what I was trying to raise was that it was good for this first set of biosimilars that we developed, right? It was good for those bevacizumabs of the world or the trastuzumabs of the world, where we had a wealth of data to calculate those equivalence margins, to calculate those which studies to choose from, what is that standard of care. Today we are in a very different uncharted territory, and you will see that, and I'm sure the division is probably looking at some of that as well as part of their discussions. We are in a very different territory here, because we don't have that wealth of data to really design that efficacy study. So I think we are really looking forward from some kind of a pathway or some kind of discussions which can help us to come up with something which is manageable. I don't think we are there to completely say that we don't need clinical studies. I don't want to go there. I think there is still, but I think they have to be manageable. I think we can't go with that same old paradigm that we have been looking at it. And in that space, I think some of these biomarker discussions would be helpful.
Any other comments to
Yeah,
add on this topic? Yeah, Leah, go ahead.
Yeah, I would just say, again, I tried to make the point in my presentation about sort of how you fit these discussions into a drug development program. And we know that typically there's multiple companies that are developing a proposed biosimilar to a given reference product all at the same time. So, they're, even if you really want to try to push the envelope and get on board with the science of this, again, that sort of lack of predictability and transparency and certainty about how to move through those conversations and pathway while you're doing drug development, it gets to be challenging because you don't have as much certainty. And as my colleague just said, if you have someone else who's taking a more traditional pathway and there's more certainty that's there, if at the end of the day you really try to push the envelope on the science and really try to dig into this, but you don't end up having what's necessary, where does that leave you? And so, there's things that I think need to come up front from regulators, not just about how to have these scientific conversations, but, more about what's being looked for in terms of the endpoints that we're talking about, how to develop that data, what is it that regulators are going to be looking for, and have those scientific conversations early, and maybe have opportunities to have engagements with regulators earlier than we do right now for development programs, and talk about some of these conceptual aspects for development program before you would traditionally, kick off your development program. And that, I think, means that we need to see some flexibility in the regulatory space of, either some different meeting types or different opportunities to have these types of engagements, so that, there is some certainty within that development paradigm once you start that, so that you don't end up kind of behind the 8 ball for time, trying to support these scientific concepts, but at the end of the day, not having the data that you need.
Yeah, thank you. Any other comments on this topic from FDA or otherwise?
Yeah, maybe I could just basically say, really mirror what Leah just said. This is exactly what EMA is trying to accomplish. And we're also on a learning curve, I must say, quite clearly, in terms of an organization, in terms of EMA and the way we are proceeding here. We're on a learning curve. But what we do want to encourage a new meeting structure, more interaction at an earlier time point, and really what we said before, have it a stepwise process, really have it stepwise in a sense of really looking at the data one set and then taking a decision on the next step. So this would really be stepwise, but the cut point would be a different one. And I think it's so something we're experimenting with right now, and we would like to encourage really applicants to come and try this, and I guess it's a mutual learning. This whole discussion reminds me a bit of the initial discussions and papers we had to write and so on in convincing the stakeholders in terms of extrapolation of indications. So this whole idea that you do a clinical study, a comparative efficacy study in one particular indication, then basically confirming what you've seen before and reducing residual uncertainty, and then getting granted the entire labels that are available for a particular product. And there was a lot of hesitancy really with many stakeholders to accept that. So why would a drug work in an oncology indication if the study was conducted in rheumatoid arthritis or such? And now it's taking this even one step further, in that we are looking in a way for the right model, a sensitive model, where you could go in a different patient population even than you have on the label, possibly in a different dose than what is on the label, with different endpoints, not even clinical endpoints, but pharmacodynamic endpoints. which may or may not have been very established and accepted for other approvals that are standalone BLAs. So I think here it's taking the uncertainty level one step further, and I really like the intervention very much in the beginning, of course, of Sarah Yim, that less data or different data or having even more of a model character may lead to a decrease in confidence. So really, this is also our task to again reinforce this is a model. We've got to get the model right. We've got to get the criteria for the model right, agree on those criteria, and through that then increase again the confidence level. And this whole discussion is also taking place at WHO right now, as you're all aware. We're revising the guidelines there. We've had a first round of public consultation. We've just worked in all these comments. There's going to be a second round of commenting coming up. And here we also address this, and we may change the title of extrapolation of indications to authorization of indications. So it's not so much the extrapolation anymore, but how do you authorize, and the level of evidence can be generated from different parts of the comparability exercise, and PD markers certainly can be one important component.
Great, thank you. That's very helpful. Isam, it looks like you had something you wanted to add.
Sure, just sort of piggybacking on what Elena mentioned about mutual learning, and also sort of what Leah mentioned about some kind of innovation around a platform or a meeting type to discuss this. It is very analogous to the Voluntary Exploratory Data Submissions Program, the VXES, which we did have joint meetings with EMA on. And really that program was established, I think, for two reasons. One is, on the FDA side, we didn't know what we didn't know. Remember, this was sort of the early post-genomic era, and there were just so many questions around how do you do high throughput, multidimensional analyses? What's the new biology showing us? How do we get ready? How do we ensure sort of scientific and regulatory readiness for application in the regulatory space? And then on the industry side, they were quite concerned about FDA overreacting. You have this very multidimensional, high complexity data, biomarker data. They didn't really want to share those data with us because of uncertainties around how we would react, spurious findings, potentially, and how we might sort of apply those to a product. And so that is why that was a safe harbor. Essentially, it was agnostic to a drug development program, although it certainly could be within the context of a drug development program. It was non-decisional, but essentially it allowed for this iteration between FDA scientists, EMA scientists. I think we may have had other regulatory agencies involved ad hoc, and pharmaceutical scientists to flush out precisely those questions. And so, a question on the table might look like, what happens when we have this case where we can't get these 5 dimensions of biomarker, fit for purpose laid out? What is if we can't satisfy those criteria, which one is more important? How do we, what is the quantum of evidence that allows us to move forward? What are the features around either the molecule or the disease context that would allow us to do that? How do we, theoretically or conceptually, what does a multiple biomarker approach look like in order to progressively reduce uncertainty? So the reason I'm raising this is because we do have precedents of how we've done this in the past. I just hadn't thought about it until, these comments were if that's a need. I don't think on the drug development side, we should assume that FDA has sort of a clear, picture on the evidentiary requirements for all scenarios, and vice versa. There shouldn't be an assumption that all the practical realities on the ground from the development standpoint are fully appreciated. And that would be an interesting mechanism to explore for further discussions.
So been a great discussion. I think further exploration of whether the benefits of bringing together a more extensive effort, governed, organized, to address these next steps and filling in the questions about new applications where you don't satisfy those 5 areas. There's certainly a lot of precedent for how to do that based on the successes, some that you're mentioning. Any other comments on this topic? Has been a great discussion about where to go next for more complex, newer biologics that are coming to the biosimilar market. But just want to make sure we've covered everything that people wanted to say on this. Yao Ming, did you have something to?
Yeah, I wanted to add a little bit to the other end of the spectrum. Abhijit mentioned touched on these very important topic, which is the analytical CQA assessment. So where we go, it potentially that analytical similarity piece can play a very big role. I think everybody is aware that the FDA's guidance articulated the stepwise approach, and then you try to stepwise reduce the residual uncertainty. And so the first step, of course, is the analytical similarity. Now we have a lot more tools in our armamentarium, so we should be able to do better. And with that, we could do a more targeted approach. Question now is, again, going back to the timeline of drug development, when you try to get things quickly out, how much do you prepare, and how detailed do you do in the analytical similarity assessment? Where is the investment, the return on investment? Those are calculations that I'm sure industry has, but in the long run, a good investment on the framework of how we shore up the residual uncertainty in the analytical similarity could really benefit the conversation about what clinical data do we need, and how we design that clinical program.
Yeah. Thanks, Yao Ming, for that comment. It reminds me that before we wrap up this session, I do want to come back to each of you and ask about, based on adding to what we've talked about already, you've covered a lot, any particular next steps that you'd like to encourage a focus on, where there's maybe some low-hanging fruit to make more progress in all these issues. Before I get to that, you all have been very efficient. Thank you in answering questions coming from our participants today. I did want to go to one more to Elena. There's a question about your view of the UK's new guidance for biosimilar approval that relies on a PK only approach in certain circumstances without PD biomarker or clinical efficacy and safety studies. Any comments you care to make about that? And others can make some too if they're familiar with it.
Yep, I think this is something that is also a possibility. I think if the product is really highly characterizable to an extent of being nearly identical, then that should be possible. We've already seen it with a biosimilar like teriparatide. So we've actually just asked for PK with or without PD endpoint. So I think that is possible. I think also given the fact that we do not have PD markers available, or we're not yet clear in terms of these 5 dimensions, let's say for the more complex monoclonal antibodies and such fusion proteins and so on. This could be a pathway, but again, we would like to have a very high reassurance on the preceding steps, as I had just laid forward. So rather than being more general in just sort of turning around the requirements, basically saying this is the rule, and only in the occasional situation we would ask for a comparative clinical trial, as is right now out for the UK. I think we still right now stay with what we've said before, but want to open up that on a product basis, yes, there could be the option to have less clinical evidence. May I also point out, I think one of the speakers before had said, well, even with a phase 1 or PK PD trial, you would still possibly need a phase 3 trial, if I understood you correctly, for safety or for immunogenicity. I think it was particularly immunogenicity mentioned. And we've actually reviewed the data, all monoclonal antibodies and fusion proteins on the market, a paper that is just about to come out. I think it will be published within the next few weeks in Drugs. And we've looked at that question. And interestingly, in terms of immunogenicity behavior, whatever you see in a phase 3 trial, you've already seen in the phase 1 PK trial. So if there's a low immunogenic product, it would show in the phase 1 PK trial and similarly in the patient trial. And if there was a change from a higher level antibody titer or lower level antibody, you would, again, it would be mirrored. And so the question, of course, is, do we really need the phase 3 trial for immunogenicity in a standard case, let's say, if there's no concern with regard to impurities, let's say? And this is a question we're asking ourselves right now, and it's actually giving us more confidence that the direction we want to take now in the future may be the right way.
You know, interesting point, especially as you're also getting more data around safety and immunogenicity from the real world use context as well. So we only have a few minutes left. I would like to wrap up, but before we wrap up, ask you all for any final quick comments on areas where you see some low-hanging fruit opportunities. I want to circle back to Dr. Woodcock's comments at the beginning of the session about how, as part of the action plan for biosimilars, FDA does have some, not unlimited resources, but is trying really hard. You've heard about a lot of the efforts underway already today to facilitate progress. Any areas that we haven't already talked about, you think some additional progress is possible that you'd like to just get on the table for the further sessions ahead in this conference?
I think one area we talked about, and Elena did mention about immunogenicity, and we talk about immunogenicity all the time. And you know, the purification technologies have become so advanced that sometimes the biosimilar product is kind of, you know, has less impurities than the novel. That's a different discussion for some other day. But I think the issue is that there is a lot of data that, you know, the regulators have access to, right? Which the sponsors don't. And I think the learnings from them, you know, what have the regulators seen in terms of which studies have failed for efficacy equivalence? I mean, you're talking about this, you know, what are those criteria? When you look at it versus, you know, I think the PD biomarker approach is important, and I think it's well appreciated. But I think there's a wealth of knowledge within FDA as well as EMA where they can look at some of these things. Why did certain studies fail? I mean, so when we look at what has been published, and that's always the fallacy, you don't have everything that is, published, which is always, the studies have been conducted. But if the regulators have access, I think that would be a good start to really understand what are we adding value? What are we learning from these studies? As Elena mentioned, they feel confident that immunogenicity is not big of an issue. We feel same way, but it's a good confirmation. But I think that would really help the process.
Thank you. Other final thoughts?
I think I would just say, we sort of separate the conversations that we have around clinical data to clinical PK similarity, and then we talk about these other aspects. And I think what we're seeing is that we need to more comprehensively think about clinical data and what's necessary and relevant. And to Elena's point, can you collect the safety data and immunogenicity data that you need in another types of studies? And immunogenicity data should always be collected in that PK similarity assessment, and maybe that's sufficient to do that. And so if we think more comprehensively about that clinical program within a biosimilar development program, you can feed in these aspects of PK, PD, and what else you need to be assessing, and have that flexibility that's there to design any studies that you're conducting to answer the question of whether there's clinically meaningful differences and really think about where you can have flexibility in that space.
Thank you. Sarah, Yao-Ming, Isam, any final thoughts?
I will say that there's a couple of challenges that have been already referred to in terms of timing of development and when we start getting questions about the clinical development program. I don't think that's insurmountable, but timing is another sort of wrench in the works. I think we also need to do a lot more sort of basic translational kind of correlation research, right? I mean, I think we do need to have a better idea of what magnitude of differences in the various assays should start triggering some concerns, because I think there's still a lot of unknowns there.
Yeah, back to the point about, it seems like a good opportunity for some pre-competitive data sharing and maybe to work out some of those ideas like Abhijit described about what regulators are seeing from across all of their experiences. And you all touched on some of that in this session. Well, I want to thank you all for a great discussion and did a nice job of teeing up some of the big issues around biosimilars and PD measures that we'll come back to during the course of the remainder of this event. For right now, though, we are taking a bit of an extended break. We're going to reconvene at 12:50 Eastern time for our next session. Thank you all very much for the great discussion this morning.
Thank you.
Thank
Bye-bye.
you. Bye.
All right, everyone. I'd like to welcome you back to session 2 for today's web event. This session focusing on leveraging pharmacology to advance PD biomarkers for biosimilar development. In this session, we're going to hear some additional detail about ongoing FDA activities to advance the regulatory science supporting PD biomarkers for biosimilars. We'll hear about the FDA's work toward an evidentiary framework for using PD biomarkers to develop biosimilars, and we'll hear early outcomes from the clinical trials that the FDA has sponsored to inform this proposed framework. Through these studies, the FDA has developed some additional experience measuring and analyzing PD biomarkers among biologic products with varied mechanisms of action. So this gives us a chance to build on some of the themes and challenges that were brought up in our morning session. I'd like to begin by introducing our speakers for this session. We're first going to hear several presentations, beginning with David Strauss, who's Director of the Division of Applied Regulatory Science in the Office of Clinical Pharmacology at the FDA. Then Jeffrey Florian, who's Associate Director in the Division of Applied Regulatory Science in the Office of Clinical Pharmacology at FDA. After those presentations, we'll hear remarks from our lead reactants, Martin Chiesti, who's Global Head of Regulatory Affairs Policy at Sandoz, and Jillian Willett, who's Senior Vice President at Avalere Health. Also joining our discussion, we're going to have Ping Ji, who's Biosimilar Scientific Lead in the Division of Inflammation and Immune Pharmacology in the Office of Clinical Pharmacology at FDA, and Stacy Ricci, who's Director of the Scientific Review Staff in the Office of Therapeutic Biologics and Biosimilars within the Office of New Drugs at the FDA. So let's get started right with going to David.
Thank you very much, Mark, and thank you everyone for joining us here today. Very excited to talk to you. And do I have permission to advance the slides? Let's see.
David, we can do that for you if you just tell us when to advance.
Okay. All right. Thank you very much. So my name is David Strauss. As Mark mentioned, I'm Director of the Division of Applied Regulatory Science in CDER's Office of Clinical Pharmacology. And I'm going to talk to you about developing an evidentiary framework to advance the use of pharmacodynamic biomarkers for biosimilars. A lot of the material that I'll cover and expand upon is contained in the backgrounder document that was posted on the Duke Margolis website in advance of the meeting. I'm going to cover the use of PD biomarkers in approved biosimilar development programs briefly. And Dr. Yao-Ming Wang covered this in the last session in more detail. I'm then going to take more time discussing the considerations for PD biomarker assessment and PK/PD similarity study design, and then give an introduction to FDA's action to fill information gaps and inform on best practices for PD biomarkers to support biosimilar development. And this will be discussed further by Dr. Jeffrey Florian after my talk. First, some key points that I'll also return to at the end, and you've heard about these some already. PD biomarkers for biosimilars do not need to be surrogate endpoints for clinical efficacy outcomes. Characterization of PD biomarkers according to the 5 key characteristics that Dr. Wong brought up is critical to assess their suitability. A single clinical pharmacology study can assess both PK and PD similarity in one study if designed appropriately. And utilization of PD biomarkers can eliminate the need for comparative clinical efficacy studies, streamlining biosimilar development. As I mentioned, biosimilars may be approved based on PK and PD biomarker data without a comparative clinical efficacy study. Thus, we can go from longer, expensive comparative clinical efficacy studies to more reliance on PK and PD data that can lead to shorter and less costly clinical development programs. Evaluation of PK and PD similarity can also have the advantage of being more sensitive than clinical efficacy endpoints in detecting differences should they exist. With regard to considerations for PD biomarker assessment, the criteria for PD biomarkers intended to support a demonstration of biosimilarity are inherently different from criteria for surrogate biomarkers used to support new drug approvals. In addition, as the purpose is to confirm similarity instead of independently establishing safety and effectiveness, a correlation between the PD biomarker and clinical outcomes is not a requirement. Finally, biosimilar development programs use PD biomarker similarity studies to address residual uncertainties about biosimilarity. And that's the unique context of use of pharmacodynamic biomarkers for biosimilars. You heard a little bit about the 5 essential characteristics of a PD biomarker: relevance to the mechanism of action, the time of onset of change or temporal profile, and return to baseline after discontinuation of dosing, the dynamic range over the drug's exposure range, sensitivity to differences between the proposed biosimilar and reference product, and the analytical validity of the PD biomarker assay. I'm going to go through these with some additional examples. In terms of the drug's mechanism of action, if we take epoetin alpha as an example drug, it binds to the EPO receptor, which is its target. You have target engagement. It then triggers a cascade to increase red blood cells. An early biomarker is an increase in reticulocyte count. Those are immature red blood cells. Later, you can measure an increase in hemoglobin level, and then you would detect and have an impact on clinical outcomes of decreasing anemia, decreasing the need for blood transfusions. So both reticulocyte count and hemoglobin level are PD biomarkers that are relevant to the mechanism of action of the drug and are candidate biomarkers for a PD biosimilarity study. Of note, while target engagement is the critical first step, it has generally not been considered an adequate PD biomarker on its own that would obviate the need for a comparative clinical efficacy study. What about drugs with complex pharmacology? These drugs can have many measurable PD biomarkers, and you'll hear more about a couple examples from Dr. Jeffrey Florian in his talk. It can be difficult to pinpoint any one PD biomarker as being definitive within the mechanism of action pathway. But this does not rule out a PD biomarkers focused approach to establish biosimilarity. And the need for a comparative clinical efficacy study will depend on the totality of evidence, combining the non-clinical data, in particular the analytical data, and then the combined PK, PD, and immunogenicity data. Regarding the temporal profile of a PD biomarker, it's important to understand this when selecting a candidate PD biomarker. Going back to the example I presented a couple slides ago, where you have an early onset biomarker reticulocyte count shown here, peaks within 10 days and returns to baseline within about 3 weeks, or the late onset biomarker of hemoglobin. And here it comes up over a longer period of time and takes longer to also decrease after you would stop dosing. So for the early onset PD biomarkers, they can allow for decreased study duration and increased sensitivity. Late onset PD biomarkers like hemoglobin, as an example, may require longer study periods. With the approved Epoetin alpha biosimilar, the program included data for early and late onset biomarkers, the ones shown here. There was a 20-day single dose reticulocyte count study in healthy subjects and a 30-day hemoglobin study that had multiple doses. So of note that in this program, it wasn't required to see a full peak and return to baseline, but the combination of 30 days, 4 weeks of hemoglobin study and a single dose reticulocyte counts study is what supported approval. However, biosimilars also can be approved based on data from a single biomarker. It is not necessary or not a rule that you would have to have more than one. And for example, the absolute neutrophil count PD biomarker was used for approval of pegfilgrastim biosimilars. What about the dynamic range of the PD biomarker? Well, PD biomarkers that achieve a large dynamic range over the drug concentrations achieved in the PK similarity study are recommended. This example, hypothetical data shows time and PD biomarker response, where a placebo and then increasing doses led to an increased PD biomarker response. And modeling of the dose response data can be used to identify the sensitive dose, which is not on the plateau of the dose response curve. So now the dose is plotted on the x-axis, it's the log of the dose and the PD biomarker response. And a sensitive dose range would be generally in this area, not on the plateau, where an increase in dose is not going to change the PD biomarker response. Regarding the sensitivity of the PD biomarker, the sensitivity of the PD biomarker to detect differences between products depends on multiple factors. One, I just covered this on the last slide, the dose. It's important to be the sensitive part of a dose response curve or not on the plateau. In addition, with regard to the study population, it can be important to minimize inter and intra-subject variability. Here's hypothetical data. Drug-free data could be placebo or just taking measurements over time of a certain biomarker, and change in baseline here, you can see there's minimal change in healthy subjects, and there's larger changes in patients, and by changes, I mean variability. And selection of the least variable population in which the PD response can be measured will generally increase sensitivity and decrease the sample size required for a PD biosimilarity study. What about the analytical validity of the PD biomarker assay? It's important to demonstrate bioanalytical validity of the assays, and this has been outlined in FDA's bioanalytical method validation guidance, and summarized in FDA's biosimilarity clinical pharmacology guidance. A fit-for-purpose approach may be applied when demonstrating the bioanalytical validity. Assays used in pilot studies may not require full validation, but assays to support regulatory decision-making, that is in pivotal PD studies, require full validation. I'm now going to transition to FDA's action to fill information gaps. You heard a little bit about the Biosimilars Action Plan in the first session. And under this FDA Biosimilars Action Plan, we are conducting applied and targeted research to fill information gaps, inform best practices, and evaluate new methodologies. This includes characterizing known PD biomarkers, where we've conducted pilot studies, 2 studies with 2 originator biologics each, where the PD biomarkers are closely tied to the drug's mechanism of action, and 1 study with 2 originator biologics where the PD biomarkers are for drugs with complex pharmacology. And this will be presented in more detail by Dr. Jeffrey Florian in the talk right after mine. In addition, in these studies, we're exploring the use of new technologies to identify PD biomarkers or assess multiple biomarkers simultaneously. This includes proteomics analysis and small RNA transcriptomics. I'll cover this in an additional slide in a minute, and further details and some preliminary results will be presented by Dr. Paula Hyland from FDA tomorrow in session 4. The primary outcome measures for these studies are standard pharmacodynamic metrics, such as specifically area under the effect curve and maximal change, and Dr. Florian will cover this more, for a primary pre-specified PD biomarker at multiple doses, either 3 or 4 doses for each drug, depending on the study. Secondary outcome measures are, again, the same standard PD metrics, but for a secondary pre-specified PD biomarker in 2 of the 3 studies. And then in all the studies, PK characteristics at multiple doses for each drug, and PK/PD model parameters after combining data from multiple doses. As I mentioned, we have exploratory outcome measures, also plasma proteomics, which is the differential expression of proteins in the blood and plasma. And we can assess many proteins simultaneously, and plasma small RNA transcriptomics, specifically looking at differential expression of microRNAs, which can be very specific biomarkers. And on that note, a little bit more detail on this slide. The proteomics, we are assessing 7,000 proteins simultaneously, and this is some data from our study with the complex pharmacology drug, just one example biomarker, time post-dose on the x-axis, PD biomarker response. You can see this temporal profile with treatment, the highest dose versus placebo, and this is raw data with box and whisker plots. And here are 4 more examples of plasma proteins that had significant changes after dosing, and there are many more than 4 that had significant changes, and this seems to be very promising for biomarker identification if you don't have well-characterized biomarkers. And again, this will be discussed in more detail by Dr. Paula Hyland tomorrow. In summary, in working to develop a evidentiary framework, we've been considering evidentiary considerations for the 5 biomarker characteristics and additional factors, including study design and details about different types of biologics. We've been performing targeted applied research to fill information gaps, inform best practices, and evaluate new methodologies. And we're very excited to be holding this workshop with Duke Margolis and all of the partners here. We have representatives speaking or participating in panels from 4 regulatory agencies: FDA, EMA, Health Canada, and Japan Pharmaceutical Medical Devices Agency. The last session tomorrow will include regulatory perspectives and presentations from and representation from all those different regulators, as well as industry. We've heard a couple speakers, we'll hear some more, and academics and practitioners. And we're looking forward to getting feedback and working together. The goal is to increase utilization and acceptance of PD biomarkers to streamline biosimilar development. And just quickly to return to the key points, biomarkers for biosimilars do not need to be surrogate endpoints for clinical efficacy outcomes. Characterization of PD biomarkers according to the 5 key characteristics is critical to assess their suitability. A single study can assess both PK and PD similarity, and utilization of PD biomarkers can eliminate the need for comparative clinical efficacy studies, streamlining biosimilar development. Thank you very much.
David, thanks very much for a great overview. And next, we'd like to turn to Jeff Florian. Jeff?
Give me one moment, please. So thank you for the introduction. It's my pleasure to speak with you today about ongoing research activities at the FDA to help address information gaps and inform best practices in regards to study design and PD biomarker analysis. For this talk, I will provide an overview of these research activities, as well as a high-level look at the different clinical studies. The remainder of the talk, I will focus on study characteristics and results from 2 of the studies before concluding with a summary. To gain a better understanding of challenges with conducting pilot PK/PD studies and gathering information to inform such, FDA conducted 3 clinical studies across 6 products. The aim of these efforts was not to focus on any one product. Instead, it was to gain experience across different product classes on the conduct of pilot studies, including challenges gathering information to inform their design, as well as sample and data analysis. 3 different product classes were considered. PCSK9 antagonists represent one, where a potential PD biomarker was also the surrogate endpoint in the clinical studies used to support approval. IL-5 antagonists are a class with some PK and PD data available, though there were information gaps in what was collected. There are potential biomarkers related to the mechanism of action that have a direct relationship with the clinical endpoint, but were not the clinical endpoint. And finally, interferon beta-1a products are a class with less complete biomarker data and limited dose exploration. While the PD biomarkers are related to biologic activity, these products have complex pharmacology, and it's difficult to determine the exact mechanism of action. An outcome of these pilot studies, we anticipate the data would help fill information gaps in these areas, and that the experience and knowledge gained helps to inform best practices and methods for biomarker selection and characterization. Clinical pharmacology biosimilar guidance outlines study design considerations for pilot studies and PK and PD similarity studies. Some of these are highlighted here and were used to guide our studies. For design, crossover or parallel designs may be used. Parallel study may be needed depending on half-life of the product, or if there are concerns about immunogenicity or carryover effects from crossover design. Selected population should be informative and sensitive for evaluating PK and PD differences. Studies can be conducted in healthy participants when possible. Drug is safe if the PD biomarker can be observed and quantified in that population. Pilot studies, a range of doses should be used. Dosing should be sensitive for evaluating differences and be at a clinically meaningful dose when possible. Same route of administration as the reference product should be used. Sampling and study design will depend on what may already be known about the PD biomarker characteristics and will inform duration of sampling to capture onset and offset of effect. PD biomarker will have to be summarized as area under the effect curve, or maximum effect, as Dr. Strauss had mentioned, compared across doses. Single PD biomarker may be considered acceptable, or a broader panel PD markers for capturing different pharmacologic effects may be needed. Study designs used in our pilot studies are summarized here. All were single-dose, parallel, placebo-controlled double-blind studies conducted in healthy participants. All studies included intensive PK, PD, and exploratory proteomics analyses for characterizing potential PD biomarkers. A range of doses were evaluated per product. Placebo arms were included to help characterize the dose-response relationships. All PCSK9 and IL-5 treatment groups had 8 participants, and there were 12 participants per group in the interferon study. PD biomarkers were selected based on review of the literature and published reviews for all products. Worth noting, the PD biomarkers for interferon beta 1A have also been recommended by EMA in their biosimilar product-specific guidance. PD endpoints follow the recommendations in the clinical pharmacology guidance. For the remainder of the talk, I will focus on 2 of the studies, the PCSK9 antagonist and interferon beta 1A studies, going through the study design rationale and preliminary observations. We use literature and other sources to evaluate the PCSK9 antagonist in the context of the 5 PD characteristics discussed earlier. PCSK9 antagonists that were looked at are both monoclonal antibodies with a clear mechanism of action. These antibodies bind the free circulating PCSK9. PCSK9 binds to LDL receptors on the surface of hepatocytes, promoting degradation of the receptor. By inhibiting binding of PCSK9 to this receptor, these drugs increase the number of receptors available to clear LDL from the blood. On this slide, I'll cover some of the rationale for the PCSK9 study design characteristics. Published information supports that the biomarkers of interest are available and could be studied in healthy volunteers. LDL-C was a surrogate endpoint for some of the indications and was selected as the primary biomarker. Other measures, such as ApoB and PCSK9, were also included in the study. Both of the drugs, a range of doses was selected up to or exceeding the lowest approved dose. The lowest doses were selected so as not to saturate PCSK9 inhibition and provide full range of doses for evaluating the LDL-C dose-response curve. There's a lot of PK and PD information available in the literature for these drugs on time course, so the focus is more around exploring variability in the measures, how to analyze such data, and exploring novel methods for identifying potential biomarkers. On the next slide, I'll walk you through the findings from the study. Pharmacokinetic profiles are shown for alirocumab on the left and evolocumab on the right. Profiles for all 4 doses from each drug are shown. Each drug was administered as a single dose at time 0 with subcutaneous injections. For alirocumab, doses of 15, 25, 50, and 100 milligrams were used. For evolocumab, doses were 21, 35, 70, and 140. Drug exposure increased with increasing dose, was overall in good agreement with previous published findings. The 2 lowest doses of evolocumab, which were selected based on PD targets, were mostly below the limit of quantification in our PK analysis. Shown here are the free PCSK9 results for evolocumab, one of the secondary biomarkers used to inform dose selection. PCSK9 decreased rapidly following drug administration at time 0, recovers as concentration decreases, overshooting the original baseline. Even though the concentration for the 2 lowest doses were below the limit of quantification, there are still clear effects on PCSK9 with those doses. 3 highest doses showed near complete inhibition of PCSK9, with approximately 50% inhibition observed with a 21 milligram dose. Shown on the right is the time course change from baseline in ApoB, another secondary biomarker. The placebo response was fairly flat. Increasing effects were observed on ApoB with increasing dose. ApoB response was collinear with the observed changes in LDL-C, which is expected based on biology. They're bound and eliminated together. The data from ApoB did not have any clear advantages of PD biomarker compared to LDL-C in terms of variability or other features because of this. Before discussing the ODLC data following drug dosing, first I'd like to mention an observation from the placebo data. Mean ODLC data from participants given placebo, shown here on the right. Dotted line shows a change of 0 from baseline. The dashed line showed the interquartile range of the placebo data, which extended down 20 milligrams per deciliter below baseline. Variability in PD data, which will be a point discussed in sessions tomorrow, and impact analysis and interpretation of the data. The larger the baseline variability is relative to the effect size, the more critical it is to take steps to mitigate variability. This can be done, such as averaging multiple baseline measures together. Variability can otherwise overwhelm the effect and observations from lower doses, particularly when deriving exposure metrics from the data. This is highlighted on this side. ODLC change from baseline is shown for alirocumab on the left, and evolocumab on the right. Dotted line is before is baseline. Placebo data is common to both figures and the same as what was shown on the previous slide. Dose-dependent changes and increasing effects were observed on LDLC in both cases. However, some of the lower doses have effects substantially within the band of the placebo noise. Also, while treatments return to baseline, none of the profiles exactly settle on baseline. This is not a lingering effect, as shown by the placebo data, which has a similar profile. Rather, it reflects variability and is an important consideration when calculating PD measures, such as area under the effect curve. When analyzing PD biomarker data, it is advised to look at maximum changes from baseline as well as area under the effect curve. We will discuss area under the effect curve assessments in more detail here. A measure that adjusts for baseline should be used, particularly for cases where the PD biomarker is inhibited or reduced from baseline. The figure on the right illustrates the area of interest, cases where the PD biomarker is decreased. The yellow highlighted area, which is the area beneath the PD biomarker curve, is not a direct measure of the drug effect and is insensitive to differences across doses. Instead, the area of interest is the hashed region between the baseline and the PD biomarker curve. That is what is referred to as the area under the effect curve for these analyses. We're discussing baseline adjusted measure here, but one can consider using a percentage adjusted metric too. That is most important when the baseline measure varies by multiple fold, which is not the case for this data. The table summarizes mean AUC measures across the evolocumab doses. There are two observations here. One, variability in the measure increases with the lower doses, upwards of 100% coefficient of variation with the lowest doses. Two, a mean summary of the PD biomarker data, even with averaging baseline and other steps, may not show clear trends. This information is better evaluated using modeling approaches to integrate data across all doses. We did that with the study data to better understand the dynamic range and sensitivity of different doses. We'll start first with the alirocumab data. Shown on the left is the dose response results for baseline corrected area under the effect curve. On the right, the maximum LDLC decrease from baseline. Y-axis has been normalized to fraction of maximum effect to allow comparisons between the assessments on the same scale. Response curves in this case decrease because this is an inhibition. PD marker decreases. Doses shown on a log scale to emphasize absolute changes in response to some of the lower doses. All alirocumab doses are shown on the curve, and we see that doses fall in different locations of the respective dose response curves. The entire span of this is the dynamic range of response. Certain portions are more useful for evaluating differences between a reference product and biosimilar. Doses not on the steep part of the dose response curve are preferred. That would correspond to the theoretical dashed green line at the bottom. But doses can also be too low, leading to impractical studies. Steepness of the curve may be higher at lower doses, but absolute changes are smaller and overwhelmed by noise. It results in a middle area that balances these two concepts. We can see that a set of doses fall within this region. It should be noted it isn't necessary to have equal sensitivity across all PD measures. Here, it may be more important to have a sensitive dose for evaluating area under the effect curve, which would reflect full drug effect. Similar assessment was done for evolocumab with similar results. Shown here, are the same pair of plots. Area under the effect on the left, maximum LDL-C decrease from baseline on the right. The plots have been normalized the same way. Rather unsurprisingly, the curves are quite similar. One of the advantages of studying a pair of drugs from the same pharmacologic class. The available data can be used to establish a dynamic range, a point where the dose response is less steep, as well as a region where the noise and variability may make it impractical to evaluate differences between products. Summarizing findings from this study, we obtained results that align with published findings for these products. Results highlight dose selection across a range of biomarkers in a known cascade. Data from various PD biomarkers was collected, LDL-C being the main focus. LDL-C baseline corrected area under the effect curve increases up to the therapeutic dose, and these values are on the steep part of the response curve. Most other examples with PD biomarkers have been stimulatory compounds or drugs with clear separation from baseline. This data highlights an example with inhibition, the importance of having doses that can be distinguished from baseline noise. Finally, these results also add to the experience of informing a PD similarity design, which is how to reduce variability in other steps. Next, I'd like to change focus to the study covering interferon products. There are 5 interferon beta products approved for the treatment of patients with relapsing forms of multiple sclerosis, with many of the approvals 2 or more decades ago. These are products of complex pharmacology, where the mechanism of action for the drugs and how it relates to the disease process is not well understood. It is hypothesized to be related to anti-inflammatory properties and the ability to limit leukocyte migration across the blood-brain barrier. But there may be multiple contributing factors. The literature describes multiple biomarkers that are altered by administration, includes neopterin, exovirus resistant protein 1, and beta-2 macroglobulin. However, there is limited dose-ranging information in the literature for these biomarkers. These are the ones with the least incomplete information available. As before, available information was evaluated to inform design elements for the study. Some potential biomarkers would require assessment in patients, but the ones listed on the previous page could be evaluated in healthy volunteers and were selected for study. Neopterin has the most complete information available, with discussions in the literature relating it to drug effect, so it was selected as the primary biomarker. MXA was also selected as a secondary biomarker, a potentially independent piece of information from neopterin. Doses included up to the therapeutic dose, with 2 lower doses selected for each product. It is anticipated that 1 to 2 weeks would capture the full PD biomarker time course. While a range of doses was proposed, it wasn't known if these PD biomarkers would already be saturated, though, particularly for some of the older interferon products. Sample analysis was also uncertain, particularly for the lower doses. Highlights some of the uncertainty that there may be when pursuing a pilot study to identify and characterize PD biomarkers. The interferon beta-1a pharmacokinetic data from the study is shown on the left. Pharmacodynamic data for neopterin is shown on the right. Doses were administered at time 0 by the approved route of administration. For the neopterin data shown at the right, placebo levels of neopterin are labeled at the bottom. 3 doses were evaluated, 7.5, 15, and 30 micrograms. For the interferon PK, day of increases in plasma levels are observed across all doses, with greater exposure for the higher doses. Similar increases were observed in neopterin across the dose levels, with about a 12-hour delay. On this slide, the neopterin data for both interferon and peg interferon are shown. Peg interferon data was followed for an additional week because of differences in pharmacokinetics and anticipated differences in the PD response. Placebo data was common to both drugs. Greater increases in neopterin were seen with increasing doses as it went from the lowest to the intermediate to the highest. We previously discussed looking at the PD biomarker in the context of the placebo response and variability. In this case, there's clear separation from placebo across all doses. There's also limited value in looking at the data's percentage change from baseline, or rather than as just a change from baseline for the dose response analysis. The next step, we analyzed the PD biomarker data as before to better understand the dynamic range and sensitivity. First, we'll show the results for interferon beta-1a. On the left is the dose response for baseline corrected, area under the effect curve. And the right is maximum dioptrine increase from baseline. As before, the y-axis is a fraction of maximum effect to allow for comparisons between assessments on the same scale. The curve in this example increases because dioptrine changes also increase from baseline. In contrast to the last example, both PD measures here have similar relationships with respect to maximum fraction of maximum effect. That allows us to identify dynamic range for interferon with respect to both measures. We're interpolating the relationship below 7.5 micrograms. As before, relationships allows us to section off a piece, approaching the plateau of the dose response, whereas a lower region, where due to biologic variability and small absolute changes, would be impractical to identify differences in a PD similarity assessment. Doses that were evaluated mostly fall within this region. This was repeated for PEG interferon, and as before, similar results were observed. Same pair of plots are shown, area under the effect curve on the left, maximum change on the right. Both measures are predicted to saturate with increasing doses. Both results are similar to one another. Available data can be used to establish the dynamic range of the PD biomarker, a point where the dose response is less steep, as well as a region where the noise and variability may make it impractical to evaluate differences. In this case, all the doses evaluated fell within the labeled region. So some takeaway points for the interferon study are listed here. First, there was limited information about the products with respect to changes in the PD biomarkers over different doses. Collected data addresses those uncertainties, showing that dioptrine has clear response across all doses for both products. This information could be used to guide dose selection and study design for a PD similarity study. First example, there was discussion around baseline variability and ability of doses to separate from placebo. That was not an issue here, if all doses for both products clearly separate from placebo. Unsurprisingly, the variability here was also lower than that example. Dioptrine PD saturates at higher doses, but the theoretic dose in these cases was below or around what was labeled as a theoretical transition point in the dose-response relationship. As this is a compound with more complex pharmacology, there may be considerations for other biomarkers as well, and similar analyses would be performed with the MXA and omics data as it becomes available. So in summary, FDA conducted 3 pilot PK/PD studies, each with 2 marketed originator biologics with the same mechanism of action. Studies were designed and carried out as outlined in existing guidance, and a better understanding and appreciation for challenges associated with such studies. Results from 2 of the studies were presented today, and a key takeaway is these studies have all helped confirm existing information or address information gaps regarding potential PD biomarkers. Studies themselves will also inform best practices with regards to design, challenges with sample analysis, and considerations for data analysis. The point highlighted was ways a PD metric may be summarized, and rationale for those approaches. It's also emphasized using modeling to integrate available information, help identify dynamic range, sensitive dose region for subsequent PD similarity studies. There is such a thing as too low of a dose, as biological and bioanalytical noise can hinder practicality of conducting studies at too low of a dose. Doses should be sensitive to identify meaningful differences. There's value in moving away from a concept of most sensitive dose. To further assist with best practices, the intent is to share this data and experiences publicly so others may use and benefit from it. It's worth reiterating that PD biomarkers for biosimilars do not have to be surrogates or reflect clinical efficacy. The aim of a biosimilar is not to demonstrate efficacy and safety, which has already been shown from the reference program. Rather, it focuses on rolling out clinically meaningful differences. The broader discussion about what is needed to address this will be tried to consider PD biomarkers beyond surrogate endpoints of the FDA studies. There was secondary biomarkers to see if there was decreased variability, where the interferon studies where dioptrine and MXA were evaluated. Dr. David Strauss mentioned there's ongoing work with all of these studies, including using novel omics methods for further biomarker identification. Some of these observations will be discussed tomorrow by Dr. Paula Hyland during session 4. I'll conclude with the same slide that David finished with. This is all part of a broader effort to facilitate use of PD biomarkers to streamline biosimilar development. It includes clarifying and discussing evidentiary considerations for the 5 PD characteristics, forming targeted research, fill information gaps, and inform best practices, public workshops such as this. The aim is not a focus on one product, but a broader discussion on expectations and challenges with using PD biomarkers in biosimilar development. And ultimately, what can be done to increase utilization and acceptance of PD biomarkers for streamlining biosimilar development. I'd like to thank many members of the team and turn it back over to you.
Great. Thanks very much, Jeff. A great summary of a lot of relevant research that FDA is doing. I appreciate your putting it back in the context of that evidentiary framework and goals too. So at this point, we've invited a couple of commenters, Martin Chiesti from Sandoz and Jillian Willett from Avalier, to kick off the discussion. Each should have about 5 minutes to provide some opening comments discussing the perspectives of industry experts and sponsors on the issues we've just been hearing about and their own thoughts on the science underlying PD biomarkers and comparability assessments and where we go from here. So Martin, could start with you.
Yeah, thank you very much, Mark. So first of all, thank you very much for the opportunity to speak here a couple of minutes about this very important topic, which is hot on the plate in making biosimilar development more efficient. And as mentioned already in the first session, and we've seen the examples which were approved already by the FDA, that biomarker can be a wonderful method or tool to streamline biosimilar development and to replace also comparative efficacy studies. From a company perspective and industry perspective, however, there are significant challenges in using this pathway. I mean, you talked about the low-hanging fruits, and I think whenever there is sufficient information in the literature available about biomarker responses, where especially also variability data are published, easy also when the mechanism of action is very clear, straightforward, including a PD biomarker. So these are examples where this approach is very helpful, and also the guidance which was provided by the FDA, I think, provides very clear guidance how to start with biomarker development. There are challenges, however, when it comes to concrete criteria. So when do you set the cutoff when a biomarker is sufficient to fulfill the application of replacing a comparative efficacy study? And the other challenge is biologicals, where there is very little literature about PD responses available. And for the second one, we've seen that often PD biomarker responses have a very low dynamic range, which makes them insensitive in many cases. And the second issue here is also about the variability of the biomarker data. And also the data just shown from the FDA about the PCSK9 and interferon pilot studies also show that even in cases where literature is available, the issue is also here with the variability, which makes the application from an industry perspective challenging in the sense that if the variability is more, or if the variability is not so reproducible, that what you see in the pilot study does not reflect what you see then in the end study. So this can affect significantly the subject number, and also the need maybe to do after one pilot study a second one to really correct for variability, making this essentially then also very time consuming and less lucrative to go this path. So in my personal opinion, the PD biomarker approach is a wonderful way to streamline biosimilar development if those characterizable biomarkers exist. But I also believe that, or I think that it is a way, or it is an option for the minority of biological assets. And for the larger portion of biologicals which we see around, especially the newer ones where there's very little literature available, the search for a biomarker overcompensates the benefit of streamlining biosimilar development. So therefore, I would also propose to look biomarker as one option to streamline biosimilar development only, and another option would be that was studied again. That if you look at the PD biomarker, it is a measure for efficacy. So it's certainly wonderful if you can do this. However, there are also other measures for efficacy, and this we can find also in the bioassay characterization of these biologicals. And this is in the second trend in streamlining biosimilar development, that it was also mentioned briefly in the first session, that if we can characterize biologicals sufficiently enough, and especially if we then have a bioassay panel available, which can characterize all the receptor events sufficiently of a biologic. And this can be done, for example, easily now for monoclonal antibodies, where we know the number of receptors which can interact with an antibody, and they can be measured. The binding effects can be measured, and also downstream bioassay effects can be measured with a very high precision. And this has also the beauty that this experience can be repeated quickly and multifold. So therefore, this provides a much more sensitive manner to evaluate whether two biologicals are functionally the same. And this, and if this can be demonstrated, then it is logical that these molecules will behave also in the clinical setting from the efficacy point of view the same way. So summarizing up, again, PD, if they are available, is a wonderful way to develop biosimilars. However, I would say in the future, we should put more hopes to this tailoring approach, where a sufficient bioassay panel can substitute for or can provide this comparative evidence for comparative efficacy between a biosimilar and a reference product.
Thanks very much, Martin. Maybe we can come back in the discussion to some of your ideas around sufficiently robust bioassay panel when you're not at all 5 of these key characteristics for the framework. Right now, I'd like to go to Jillian.
So thank you. Great meeting. And it's always fun to look at great science. Absolutely no question. So the opportunities for PD, exciting for sure, but they're also somewhat endless. So I'm going to try and return perhaps to the principles necessary for sound regulatory science as the basis for efficient biosimilar development and licensure. So I'm going to take a little bit of a step back and address a few of the comments made early on. So once a biologic is licensed by FDA, I would suggest complexity is no longer an issue, because the agency is then confident it can be made, and this will include the to-be referenced biologic, can be made consistently, including after manufacturing changes using established regulatory science. So this is an endorsement of kind of where we've got to. Make a note that all biosimilar studies are comparative, but we need to particularly focus on clinically meaningful ones. We're not looking for any difference for the sake of finding a difference. Residual uncertainty, another term we use extensively, also means defining what you don't know and designing your studies to answer those precise questions. So the goal overall of biosimilars compared to their reference in their development is to confirm sufficient likeness such that you have this expectation for the same clinical outcomes. And of course, that includes totality of the evidence. So my key point here is we're not just looking for differences. We're looking for clinically meaningful differences at the dose for which the reference product is used. So the key questions become, what is unknown after the initial biosimilar development? which is analytics plus PK and maybe some of Martin's functional studies. PK is generally agreed to be the most sensitive clinical assay available. Indeed, Dr. Woodcock said as much in her introduction. So what is outstanding? And are those questions that would be answered by PD or indeed comparative clinicals? And I'm suggesting that PK has already got you a very, very long way to knowing what the differences are. And indeed, we rarely use it when looking at comparability. So I'm agreeing that PD studies are interesting and they may be of value in lieu of clinicals, if indeed they can be accepted by the FDA as adequate, even if not validated. So there's an important discussion. The timelines were important, as discussed earlier, as to how you mitigate regulatory risk. But I'm struggling to see their value in terms of scientific validity. What new conclusions do they offer comparatively such that they tell you something you don't already know from your PK? And if they lack such scientific validity, then they lack ethical validity too. And this is my worry, is we need to be affirmatively knowing what each of our clinical studies is going to tell us that is new over and above what we already know from being highly similar, from having functional assays and having matching PK. And we shouldn't be doing clinical studies unless we know that there is something that is new to be told to us from doing those clinical studies. So it's not that I don't like PD. I think there's some really good stuff to be found. I'm just not sure it's got that much value for biosimilar development as opposed to, say, originator biologic development or use in manufacturing changes. Thank you.
Great. Thanks very much for your comments, Jillian. Definitely want to come back to some of those points as well. First, I would like to introduce Ping and Stacy from the FDA. As a reminder, they'll be assisting in our discussion as well. And then a reminder to all of you, if you do have any questions or comments that you'd like to introduce to the discussion, the rest of our session today, please put those into the Q&A function. Ping, Stacy, let me turn to you all at this point. Any questions or any comments that you all would like to add?
Hi, thank you. Thank you, Mark. And thanks to the other speakers today. I would like to just sort of bring things back to a point that most everybody touched upon, and that is the fact that it's really hard to make generalizations when you're talking about biological products and therapeutic proteins. You know, we like to think that the same set of data would be needed you know, for all these products to support a demonstration of biosimilarity. But as we know, they have a great diversity in their size, molecular complexity, in, you know, even their manufacturing, how they're used clinically, the diversity of indications, whether they're systemically administered or not, whether PK is even relevant, or PD are going to be even relevant. So it's important that we think about the data in really in the context of a reference product class that would be, you know, that's needed. And even then, the type of comparative analytical data that we can collect, though very precise for, you know, the majority of product quality attributes, there are some data that, you know, no matter how careful the manufacturing process is designed for a biosimilar product, there may be some small variations in a given product quality attribute. And so it's those variations that raise this residual uncertainty that will guide the type of clinical data that can sort of provide a sensitive measure of whether these things could lead to a clinically meaningful difference. And, you know, I think there's a lot to consider regarding sensitivity of clinical endpoints. Many have said today that the PK is the most sensitive, and that's certainly been articulated in guidance. And then it's sort of debatable, perhaps, according to a reference product class, whether a PD biomarker, if they do exist, their sensitivity, but also the efficacy endpoints that are used for clinical studies to support these products currently or that are being contemplated. And I think there's going to be discussion of this more tomorrow, but how sensitive are those endpoints too? So, the biosimilar program in the US is, while it's 10, 11 years old, it's still fairly, as Sarah pointed out earlier, in its growth phase. So as we get more information and more comfort with the types of data that we can collect currently, we do hope to streamline development programs in a consistent way that does lead to regulatory certainty for developers, so that they understand, what data ultimately would support approval of their product.
Great. Thanks very much, Stacy. Ping, any comments that you'd like to make at this point?
Yeah, okay. So I will probably only make a general comment, and I echo what has been said and presented. From regulatory perspective, the current guidance actually provides a flexible approach. I mean, in terms of like a stepwise approach and along with totality of evidence. And these are just to assess residual uncertainty during development at each step of the way. And this sort of provides product-specific flexibility to establish like a no clinically meaningful difference. And this flexibility also applies during drug development, as the assessment of one element, for example, the structural analysis at one step can influence the decision about the type and the amount of subsequent data for the next step. But I mean, the question here we're talking about as far as what should be used as evidentiary framework, and this one is really boils down to what is the residual uncertainty, and if no clinically meaningful difference is established. And here, I mean, as already been presented, when PD is providing a useful tool because if it relates to mechanism of action and may also satisfying the 5 characteristics. So, I mean, I think at this point, I would like to treat PD as, what other people have said, like one useful information to help drug and biosimilar development. I mean, certainly I agree that there are obstacles, but I think it can be one option. I mean, if information alone and if information is available, if it certainly satisfies 5 characteristics. I mean, but I do see more opportunities ahead, given that new technologies and also awareness about all the stakeholders. I mean, through either education and other different venues. And also, actually, we are, accumulate our knowledge and our experience about biosimilars. And so far, we have 10 years of experience. I mean, over time, we'll have a lot of experience. And this is just what we have for insulin products, for which we have a lot of experience, and given the structure and given what we know about insulin, we make regulatory decision on what would be the regulatory pathway for insulin. And the same thing could apply to others. I mean, given this, over time, I think, at least I hope that we can achieve like a sort of abbreviated biosimilar development. Of course, we have to think that it should not be at the cost of lower regulatory standard. Thank you.
Thanks, Ping. Yeah, the biosimilars program, while as you all said, still a teenager in some ways, is definitely getting more experience and growing up, and this all helps. I'm wondering if we could start, David and Jeff, I go back to you for comments on some of the feedback that you got from Martin and Jillian, and maybe starting with Jillian's comments, pretty basic about how far PK can take you in terms of assisting with scientific validity and being a sensitive marker. And if you all might have more to add about when any differences of views about that, and when the PD biomarkers are particularly likely to be valuable. And then I want to go to Martin's comment.
Yeah, I could. This is David. I could make a couple comments first, and good to hear from Jeff as well. And thank you, Martin and Jillian, for your comments and thoughts. We really appreciate that. We agree that PK is almost always the most sensitive clinical study measure, and it's critical to assess, and not necessarily saying that at some point that would, I know, Jillian, you've written about, well, you wouldn't need to do more than a PK assessment. And I think, every product needs to be assessed on its own that comes forward in a biosimilar development program. I think some of the comments about what does a PD biomarker assessment add, certainly you can have PK similarity, and then the effect of the drug on the body, though, could be different, and it could end up with a different clinical endpoint or clinical response. And if we can have PD biomarkers that can just be added on to the clinical PK study, you're not actually having to run independent similarity studies, and then may help push us over the edge to saying you don't need to progress to having a clinical comparability efficacy study. So I don't know, I'll pause there, and maybe Jeff, you would like to add to that?
Yeah, Jeff.
I was
think you're on
muted. I
mute.
apologize. Yes. So no, a few things to add to what Dr. Strauss said. So I think overall there's certainly long-term alignment and agreement trying to keep advancing this. And all of this comes back to, let's say, that slide that Dr. Sarah Yim presented. And if we think about maybe what was outlined there, going more on PK in vitro, that's going very, very far down the chain. But we're still in the teenage years. We're even trying to move away from comparative clinical studies and getting experience with PD biomarkers and those effects. That's going to help us sort of crest that top. And as experience accrues, and I think that's going to be what helps us get from maybe we are now to a next step, and then ultimately maybe to what Jillian and Martin were outlining, something that can utilize and leverage that information from the earlier analytical assessments. Like right now, we don't know how that ties in with the information. So there needs to be sort of continued accrued experience to help us understand that.
Great, thank you. And Martin, I see you've got your hand up. I did want to have a little bit more discussion on the points you raised about what to do when you can't meet the 5 elements of the framework. But if you all want to get in now, we can discuss this a little further, and then maybe back to that topic further. So Martin?
Yeah, I just wanted to make the comment that PK is measuring something different than PD. So PD is really a measure for the function of the molecule. So the future would be then PK, sensitive PK, with a combination of a sensitive panel of bioassays, which we have for many molecules already today. So just as a brief comment.
Great, thank you. And Jillian?
I would just like to add, I think we've got 25 years experience because we developed comparability in 1996. FDA led the world with what are the role of essentially analytics, highly similar, became Q5E. So I think we shouldn't undermine our understanding, not saying it's perfect, of biologics. And also by using those same comparability principles, as indeed EU did, going through to their first approval in 2006, albeit that was Omnitrope that FDA approved earlier as a B2, there is greater collective experience than started March 23rd, 2010. And it is global, and we can share that experience, I think, around the world that's useful to everybody. I think regulatory reliance, as we've learned from COVID, is increasingly important. So the reason we said PK in our recent paper was we actually looked at all of the biosimilars approved in Europe, US, Canada, Australia, and found if they had matching PK, they were going to be approved as biosimilars. And we didn't need to have waited for the comparative clinicals. So I'd just like to put into the conversation that time matters. Time matters to competition, time matters to patients, and time matters economically, as well as cost of investment to the number of competitors you have accessing the market. So not to throw the kitchen sink in here, but there are a bunch of other elements, consequence on time. And we don't just get to wait if the science is there. So going back to comparability.
Yeah, just to follow up a little bit on this, Julian, we had a bit of discussion in the last session about some of the European reliance, or at least UK reliance on PK only. I think that my takeaway from the discussion then was, yeah, well, we can potentially get there, but we're still gathering evidence. And it sounds like you think we should already be there for a broader range.
And I would like to know, it just happens to be I have a similar accent to those at MHRA, but I don't know their reasoning any more than the rest of you. I'm bona fide American as well. They think I sound American. So do we think they're being irresponsible? Or are they merely a stepping stone towards a European approval and ultimately a US approval, and we end up delaying access in the US? And I think as a matter of drug pricing, not to put everything on the back of biosimilars, but the expectations are so high, and we have to work out how we fulfill that access affordability for US patients too. I'm not going back to the UK to get my biosimilars.
Fair enough. David, Jeff, any further comments you want to make on this?
Yeah, I'll add, I think we also heard in the first session from a couple speakers about we need to be bringing along the clinicians and practitioners in addition. And I think, we've gained, the whole community has gained a lot of experience, and the sort of general at face value, the initial reaction is you want to see a clinical comparability, efficacy type study, but to jump completely from there to PK only data, would we be getting even if the science is there sort of buy-in from the broader community? And I'm not saying the science is there yet, it's just a if. I do think that being able to leverage new technologies that can rapidly identify, characterize PD biomarkers, and such that you can do an add-on to a PK study that's already being that has to be done, that can add a lot of value. Because even if for most cases, if you've done your analytical similarity and you've done your functional assays in the laboratory, and you get clinical PK similarity, having the extra confidence of having a PD measure or measures in humans can be quite reassuring and that yes, this is really working as intended. So yeah, that's all I have for now.
Great, thanks. I appreciate this discussion, and you can see that this is going to develop further as experience and policy making goes forward, both here and in Europe. And as Julian said, there is a lot of pressure around affordability of biosimilars, as well as a lot of need around education, engagement of clinicians and patients for confidence in these steps. So great discussion on those points. Now let's go back to Martin's comment, which is more in the opposite direction. So there's some interest in doing the PD biomarkers, but you're falling short of the framework. Or Martin, you also talked about cases where it may not be clear if the PD biomarker performance is quite good enough in terms of variability, et cetera, to be used. So what do you do in that case? David, Jeff, any comments on that?
Well, I think the way the guidance, the clinical pharmacology biosimilars guidance is written, it's fairly flexible, and we talk about the 5 characteristics and how I think you brought this up, Martin, if I remember. And if you do, we've stressed that it doesn't need to be a surrogate endpoint. The biomarker doesn't need to be a surrogate endpoint for a clinical outcome. But if there is data to support the link to longer-term clinical outcomes, well, that's beneficial if it exists. And certainly, you may not need to. There's trade-offs of these sort of more pharmacology-focused critical characteristics. And if you have links to clinical outcomes and uncertainty in whether a biomarker is going to be good enough or not, that's where encouraging, coming and talking to FDA or other regulators early in the process and getting feedback. I think at FDA we want to be looking at innovative approaches, and so I think those are all things to consider.
Thanks. Any further comments on this, or Martin, any follow-up?
Yeah, one item is really the expectations for the biomarker. We're talking about future frameworks, so analytical plus PK only. If it's not accepted yet, then certainly the PD must be sensitive. Otherwise, there is no meaning in that. But when you look in the future, and Dr. David, as he said, it's providing more comfort, so to say, if there is efficacy confirmation also in a clinical setting, if it is rated as kind of supportive information. So this could be also a way in the future, so that from the hard facts, the physical, chemical, and then the confirmation of functional comparability is done at the bioassay panel together with PK for the drug distribution. And then as an add-on, a kind of descriptive biomarker where the criteria can then maybe a little bit less stringent, so to say, because this is the issue we have in the industry, that for many biologicals, it's so little information about the variability of the biomarker, whether you have chance findings, how big is the subject invariance, so that the risk is really that what you see in the final study is not what you think based on literature or your pilot study.
Okay. Yeah. So thanks for that further comment. Let me maybe take this in a little bit different direction. It's been great discussion so far, building on the presentations, but we did start with kind of an overview of some of the regulatory science research activities that FDA is undertaking as part of the Biosimilar Action Plan to try to help accelerate and increase the use of validated PD biomarkers for biosimilar development. And this may be a question primarily for Jillian and Martin, but it'd be great for FDA colleagues to weigh in as well. What do you see as some additional regulatory science activities, further studies like the ones that FDA talked about today that might be helpful for building out the effective use of PD biomarkers?
Well, I think there is this global context. If we keep saying, even if I don't buy it, if we keep saying the FDA is behind, what are we learning from everybody else? What are we learning from global use? I mean, the good news, I think, is that all the biosimilars have panned out. All the biosimilars approved in the highly regulated markets have done exactly what they were meant to do. Extrapolation has turned out to be just fine. We had some interesting sort of different ways of looking at things, maybe in Health Canada with splitting indications. But how can we collectively pool some of the experience so that each regulatory authority doesn't feel a need to be quite so repetitive. And in this, I would throw, for example, another paper that we did, as Nobody's We Can Do papers, that said that if you have your reference product approved worldwide based on the same clinical studies, it is by definition the same reference product. So there is a capacity to be pooling data to maybe we call it real world evidence, maybe we call it something else. That are there levels that we can use that collective experience to responsibly and accountably build on biosimilar dossiers so they're not 1 million pages. I mean, seriously, guys, we've got to get this system more efficient. And reproducing things and filing more and more data that then FDA and everybody else has to read is not going to get us more products more quickly. So I guess my theme is both there's this breadth of data out there globally, but how do we focus on what is decisional, what is actionable by the agency in a manner that you can manage? So not to take carte blanche there, Mark, but
Well, that is a further step, and I think there's some relevance within the tighter topics today around PD biomarker validation, where, as you said, similar activities, data being submitted around the world, or there's some more effective ways to bring that together, integrate its analysis, and incorporate it into moving faster and advancing biosimilar. So I appreciate the point. Martin, any suggestions you have around further applied research, that regulatory science research that FDA could support to advance PD biomarker validation?
Yeah, I would say 2 ideas. I mean, first of all, this workshop is, from my perspective, simply terrific because it really combines in today and tomorrow the current knowledge and state of the art of biomarker and biosimilar development, also together now with these new FDA-sponsored studies. I think putting those data out there in the public domain together with the discussion, this would be tremendously helpful to as a kind of baseline for further thinking. And the second point is look more also on innovative product development where PD responses are measured. There is also a lot of research ongoing on PD panels, which can support also new product development, and look at those where there are occasions where also in the future biosimilar can benefit from that.
Great, thank you. And I'm sure if any of the FDA colleagues, anything you'd like to add or highlight about further directions building on the work to date?
I would like to just first touch upon something that Jillian mentioned about sort of 2 points that get at sort of the sort of strictures that we as a regulatory agency have with regard to the proprietary nature of data for the reference product and for any applicant's submission. So while we have knowledge of the manufacturing of products that are licensed in the US, we don't have that knowledge of the products that are approved outside of the US, unless that information is provided to us by the license holder. And secondly, and just to touch upon your concept of that we have all this experience from comparability data, it's important to keep in mind that for within a licensee's application, they make a manufacturing change, they submit data to support a comparability assessment. Those data are about their own manufacturing process. It's a different paradigm than biosimilarity, where the biosimilar manufacturer doesn't have the same knowledge of the exact manufacturing process that the reference product uses. And so they back engineer their process to try to match, product quality attributes as best as possible, but it's a difficult, complicated endeavor. So, and then to get to the the concept just in general about the types of data that we, the types of regulatory research that can be done. I mean, certainly within the agency, we can, we have that view across different reference products, different biosimilar programs. And we can look at the data that is submitted to us and try to use that to inform decision-making going forward. So if we realize that, perhaps certain assays, certain studies are being conducted that really are not useful for regulatory decision-making, we can tell that going forward to sponsors. And we have done that in the context of biosimilars, for sure.
Great. Thank you, very helpful. Jillian, you had a comment?
So just to clarify, when we published the paper on the clinical studies, say for an EU and US approval being the same, that's entirely public domain. So it's absolutely in the public domain that a given product approved in Europe, a given product approved in the US are based on the same clinical studies. So I'm not suggesting you use anything trade secret confidential. I would also say anytime a biosimilar has been approved, the same biosimilar, in the EU and the US, it has confirmed that those two reference products are the same. It has itself formed an analytical bridge. So to the extent that FDA has allowed an analytical bridge rather than a single standalone 351A reference product, all I'm saying with the clinical data is we're providing a clinical bridge as well that's already in the public domain. So let me just be clear, I'm not saying FDA should ever use anything trade secret and confidential. However, the collective experience that we have on the originator products is at least 12 years, if not 30, so that we can certainly apply that collectively to our understanding. So just to be clear, no use of trade secret confidential information would be expected of the FDA or any other regulatory agency. Comparability, of course, is public in Europe. That those changes have occurred, and then the matches between the most recent version of the reference product, ergo the US product has gone through essentially the same changes too. So just public information.
Thanks for that clarification. David, I see you've got your hand up.
Yeah, so I was gonna further respond to your prior question, Mark, about additional areas of regulatory science. And so I touched upon it in my talk, and I believe Jeff mentioned it briefly as well, but we did not, but more details will be presented and discussed tomorrow. The area of where we've gotten some new original data from our studies, in particular the use of technologies that can screen a series of, or many protein potential biomarkers, and that you can do a pilot study and assess, across a potentially thousands of proteins that could be biomarkers, and figure out what are the most promising ones, and it can be used for biomarker identification. We are just getting preliminary data on this now. Also, the potential that you could actually use a technology like that and say, you don't have to have a perfect match on any one biomarker, but you'd actually do a similarity study with an assessment of multiple biomarkers, and there'd be some type of statistical assessment or that you're having a similar signal. And so that's science of how you could leverage technologies to assess multiple biomarkers, and what is the sort of analytical, computer analytical method to, could you establish similarity based on that? Those are some things for consideration. And yeah, we didn't touch on them as much in this session, but there'll be some more tomorrow. And I think we'll be hearing from some academic and researchers from universities of their thoughts towards these types of areas as well.
I think that's a great segue, David, connecting today and some of these ideas for next steps that we've talked about to some of the discussions about further research and further opportunities that we're going to come to tomorrow. So if everyone is looking forward to those sessions. We are almost out of time here. You all have continued to do a great job of those of you who are our participants or in posing questions or making comments, and our panelists in answering them. Just a couple that I wanted to take. Gary, your comment about the US being different than Europe in terms of how health insurance, social insurance works is clearly some important differences there. That's kind of outside of scope for this meeting. This is about similarity and demonstrating similar clinical efficacy and safety across the products. Another part of the FDA's action plan on biosimilars involves addressing competition with the goal of improving access and lowering costs, including some important collaborations with the FTC. And there's a lot of work, obviously, outside of the FDA's domain related to how we can improve access and reduce costs through a vibrant biosimilar market. Jillian and Avalier have done a lot of work on that. For those of you who are interested, at our program at Duke Margolis, we also have a number of more insurance and payment focused projects, but that is kind of beyond the scope for here. One question that I did want to see if any of you all had brief comments on, I think, David, you may have answered a couple of these already, relates to variability among patient placebo PD biomarker measures, and does that, the one that I'm looking at from Anonymous, is does that, especially even if it's only at low doses, help characterize patients who might show low versus high effects or have differential effectiveness in other ways.
Well, I can add a comment. Anyone else, please add. Yeah, so some data was presented by Dr. Florian, by Jeff, and from the PCSK9 studies, and showing there was some variability in placebo, but whether the variability is sort of going to greatly affect your ability to run a biosimilarity study with a reasonable sample size all depends on the signal of when you're giving a drug, what is your biomarker response. And I just note that, yeah, in that study, we specifically chose some doses that were very subtherapeutic, went down to very low levels that were more, and so the bottom dose would have gone into the more variable range. But if you go to the clinical dose, or even a little bit below the clinical dose, it has a signal that is much higher than the variability, and that wouldn't pose a problem, and you could have a similarity study with very reasonable sample size. So it's all a signal to noise, that you need to take into account. And I believe Martin did mention that in some of his comments. Knowing the variability is important, and if you don't know variability, that is where some type of pilot study, and it doesn't necessarily need to be an extremely large pilot study, maybe warranted.
Martin, you got a comment? Thanks, that was very helpful, Jeff. But Martin, a quick comment on this too.
Yeah, very quickly. Also, cannot resist to say, beside of looking for biomarker options for biosimilars, I think one of the future policy research which could be done by Duke, or what I would recommend, also using this MHRA approach. So this functional characterization plus PK, also looking at this with a certain, or with the same, I would say, intensity as an alternative, so that I think it brings in the balance and makes, I would say, the reaching the adulthood of biosimilars regulation, I think, more quickly.
Thank you. A nice further comment to end on. Or actually, we are about at time. Any quick final comments from any of our panelists, discussants? It's been a great session. All right, and as David said, more coming tomorrow. So we have covered a lot today already in this session, reviewing some of the FDA's recent science in terms of developing PD biomarkers and applying that framework for their validation. Also, some ways and ideas to push the envelope on extending that framework for accelerating the availability of safe and effective biosimilars. This was on top of a really good previous session where some of the key themes coming out of that included that regulators have an opportunity and a lot of vehicles for continuing to develop, communicate improvements in our state of knowledge, and eventually clearer guidance and standards around acceptable PD biomarker data around growing up this process for bringing biosimilars to the market as efficiently as possible. And that process is going to be iterative and really highlights the importance of discussions like this one between all stakeholders that are affected by further FDA regulatory policy actions in this space. So sponsors, clinicians, patients, very importantly, as well as experts, and really appreciate the time and effort from the FDA to engage in these kinds of activities. But there is opportunity for both more research and continued reflections around the global experience with biosimilars, which in a number of respects has been growing up for longer than it has here in the United States. Now, we're going to come back to some of those issues tomorrow, especially around further developments in the science. That session, day 2, is going to start again at 10 a.m. Eastern time. Then we're going to hear a number of presentations from the FDA, as well as from academic experts, industry scientists, and others on this developing science. And we're going to spend some time talking about next steps, where to go from here, building on all of these discussions. You'll be able to access the meeting tomorrow using the same link that we used today, and hope to see many of you back here. In the meantime, special thanks to all of our presenters, panelists, discussants, all of you who took the time to be here and contributed questions and comments. Really appreciate the experience, and we hope you have a good rest of the day.